advances in neuroscience, and also translation, or the beginning of translation of that work to the clinic and to the human neuroscience. So this is great, but at the same time, these neural circuits operate and their function has to be read out by the dendrites of the neurons of the brain. No? And if you look at what's going on with dendrites, the textbook example that it's essentially spread out everywhere is that dendrites are integrating excitatory and inhibitory inputs. They just sum them up, and if you reach a threshold in the axonal initial segment, you generate an action potential, and that's it. That's how single neurons compute. No? And this is essentially the same model that we've had since the 1940s, since the McCloskey and Pitts even. No? And but there's been a problem with this model of how neurons work. And that started to be apparent with the first intracellular recordings from dendrites carried out by Sugimori and Ginas in the brain slices of cerebellum in Purkinje cells, and they showed that these dendrites have active properties, that they have spikes. So there goes the model for integration of circuit information as excitatory and inhibitory inputs in the soma. No? So this was not just the Purkinje cells in pyramidal neurons. The Zachman lab, using whole cell recordings from dendrite, also confirmed that these dendrites of pyramidal cells in the cortex, so these are the archetypical neurons in the brain, are also full of active properties. And this was also independently discovered at the Tank lab by the introduction of 2-photon calcium imaging of dendrite that showed that there are electrogenic zones in pretty much all the neurons. No? So we have a little bit of a dissociation. On the one hand, there's circuit neuroscience. On the other hand, the dendrites are doing something much more interesting or more complicated that many circuit neuroscientists believe. And also, circuit neuroscience is generating a lot of computational work. Computational neuroscience is a new field that's really arising from all of this work. And for most computational models, the dendrites, the neurons, are essentially just integrators of excitatory and inhibitory inputs. So the purpose of this meeting is essentially to deal with this dissociation between two fields and try to connect them together. No, from the one hand, circuit neuroscience, on the other hand, dendritic function, dendritic biophysics. No, and why do we care about that now? Well, I wanted to actually share with you as a teaser a piece of data that will probably impress upon you the importance of understanding how what dendrites are doing. So these are data using one of these new methods from the Brain Initiative, a genetically encoded voltage indicator. In this case, is ASAP3, expressed in vivo in the dendrites of pyramidal neurons in the mouse cortex. And this is data actually came from my own lab, from the work of Victor Cornejo, a former postdoc. But so this is what these dendrites are doing. So in pseudocolor, you can see the voltage in these dendrites and in dendritic spines. And in the inset on the lower left, you have the intracellular recording from the soma. And if you compare both things, you say, well, sometimes when the soma fires an action potential, the dendrites all light up, and that was consistent with what everyone believed as backprobating action potential. But what's interesting is what happens the rest of the time. Sorry, there's all kinds of interesting things going on, and we have no idea what's going on. No? And this essentially shows you, I'm going to just play this again, that dendrites have a rich physiology in vivo, full of small areas of potentially electrogenic properties. No? Little patches of dendrites are jumping in and out. Spines come online or not. And at the same time, what you can record in the soma does not do justice to what's happening up there in the roof of the cell. So essentially, that's just an example of why we need to worry about dendrites because and why neuroscience as a whole have underestimated what dendrites are doing, and we haven't really connected circuit neuroscience with what we've known since the 1980s about the electrical properties of dendrites. So most likely, all these responses that you're seeing in dendrites have to do with whatever is going on in the circuit. And we better understand them. Otherwise, until we figure out how the dendrites work, we're not going to understand what these neural circuits are doing, what the brain is doing. Okay, so this is the problem. This is why we organized this meeting. And in this problem, the good news is that NINDS came to the rescue, the National Institute of Neurological and Disease and Stroke from NIH, launched a program of new centers called the Combine Program to put together a small teams of people to study fundamental questions in neuroscience. And I represent a team that was awarded one of these grants to study dendrites, essentially to understand what dendrites do. And our dendrite group, which is composed of Yayita Basu at NYU, Ellie Navidi at MIT, Jeff Lichtman at Harvard, Michael Lin at Stanford, Idan Segev at Hebrew University of Jerusalem, and our own group, that used the lab at Columbia. What we're tasked to do is to study the dendrites of only one cell type of pyramidal neuron in the mouse visual cortex. And the plan is to go at it from many different angles to completely reconstruct its ultrastructure, its molecular receptor composition, to understand the voltage dynamics during a computational task in vivo, to patch them and find out what's going with the mechanisms of the electrogenesis, how is that control, to generate new tools as voltage indicators to study these dendrites in vivo, and finally to put it all together in a computational model that takes into account these electrogenic properties of dendrites. And as part of our grant, we will deliver to the field a database, a complete database. We hope it's complete, accurate, and permanent in the Brenner style, on the dendrites of at least one cell type in the brain. At least we'll look at it carefully from all different angles. Hopefully this should help us understand what dendrites are doing by having a complete database. Us and just everyone else in the field, the plan is to share this data with all the field, and we're open to collaborators. And as part of this outreach to the field, we're organizing an annual meeting, and this is the first of our annual meetings that hopefully could serve as a watering hole for the community of people around the world that cares about neural circuits and cares about dendritic computation and single neuron computation. So with that, let me just jump the program that we prepared for you today. I'm going to put the program, here we go, in the chat so everyone can download it. We're going to start with a session with 3 speakers focused on the circuit, how circuit integration. These are among the top people in the lab that care about neural circuits from different angles, from the angle of cell types, from Hui-Seng, from the angle of how dendrites, or what these neurons in the circuit do, Georg Keller and Jerry Chen. And this panel, this will be followed by a panel discussion moderated by Elin Divi from MIT. Then we'll break for lunch or a quick bite, and we'll have a second session in the afternoon in New York that will be focused on dendritic integration. Again, another 3 fantastic speakers. Michael Hausser from UCL, talking about single neuron computation. Matthew Larkram from Humboldt in Berlin, understanding how dendrites integrate information in the cortex. And Panayota Poryatsi from Poirasi from Crete, who will discuss computational models of dendrites. And this second part of the meeting with the panel will be moderated by Yayita Basu from NYU. And I think with that, we hope that you enjoy the program and the discussions that will arise from this. I wanted to thank the Chen Family Foundation and the Neurotechnology Center for the support for this meeting. And I'm going to pass the mic to two program officers from NINDS that are generously supporting this meeting as they are supporting our work. Karen David and Corey Kelly, and they will essentially say a few words about the combined program and launch us in our task to understand how dendrites work and inaugurate the meeting. So Karen, you and Corey, you have the mic. Thank you.
Thank you. Thanks for having us. Hello, everyone, and we're looking forward to learning a lot today. I'm Karen David, Program Director for the NIH National Institute of Neurological Disorders and Stroke, NINDS, and the Brain Initiative. And we're very pleased to be part of the launch of this RM1 award on neuronal integration from circuits to dendrites. And this project is one of our first cohort of this new team science program at NINDS called the Combine RM1 program. And Combine stands for Collaborative Opportunities for Multidisciplinary, Bold, and Innovative Neuroscience. This program is made possible through a team and institutional effort at NINDS. Some of the key staff are attending today. My co-lead Corey Kelly, who will speak next, and our program director Ben Chern, who handles this topic at NINDS. And this program was brought about by our intent to promote and optimize collaboration. And this program is a product of our analyses of the challenges and opportunities of team science programs at NINDS and the Brain Initiative. And our vision for this program is summarized by a quote from our NINDS director, Dr. Koroshetz. "We are stronger together through interdisciplinary team science." And indeed, team science is particularly important to neuroscience, given the complexity of the nervous system and the challenging questions about health and disease. No one person or lab is going to have all the expertise and tools needed to pursue challenging goals. So we're very excited to see your team share with the community today the promise of team science. And we look forward to being part of your journey as a team as you combine your diverse expertise to challenge existing paradigms and to discover new concepts and techniques related to dendritic function. And next up is my co-lead for this combined program, Corey Kelly.
Thank you, Karen, and thank you everyone for joining today, and thank you to our host for Neuronal Integration 2023. As Karen mentioned, my name is Corey Kelly. I work in the Research Operations Branch at NINDS, where we work to align and optimize what we're doing and how we're doing it to achieve the NINDS mission. And along with Karen, I'm a co-lead on the Combine program. Our Combine program is created to utilize the power of team science to achieve the most ambitious goals in neuroscience, to achieve transformative science, a paradigm shift, a revolution in our understanding of the brain. I often find myself going back to Thomas Kuhn's structure of scientific revolutions, or structure as it's commonly called. Kuhn wrote that during revolutions, scientists see new and different things when looking with familiar instruments in places that they've looked before. It is rather as if the professional community has been suddenly transported to another planet where familiar objects are seen in a different light and are joined by unfamiliar ones as well. And there's been a great many paradigm shifts in neuroscience through the course of history. I myself was first drawn to neuroscience through classical debates on the duality of the brain, Aristotle, Plato, Descartes, Locke, Aquinas, and how far normal science has come since the age of antiquity. Galvani's discovery of electrical excitability, Golgi's staining methods, Broca's theory of brain structure and function, Katz's modeling of the synapse, Penfield's cortical homunculus, or Columbia Nobel laureate Eric Kandel's work on signal transduction and memory in the brain. Yet, despite all these great works that we stand on the shoulders of, neuroscience in the brain still remains the great final frontier of biomedical science. And the Combine program was established for those investigators willing to take the risk of looking beyond normal science, pushing the boundaries of our understanding, and achieving transformative science, achieving this paradigm shift, this revolution in how we see the brain. And historically, the most transformative science has been basic science discoveries so fundamental that they shake the bedrock of our understanding, generating a shockwave of discoveries as it proliferates through the scientific community. And this project and the work being done here is the true embodiment of that spirit. For over 2 centuries, our community has worked on the premise that the soma is the unit of integration in the brain, and that the dendrites were simply receivers of synaptic information, funneling this information through the soma and the axon. But we now know that the dendrites have a key role in information processing, owing to their capacity to influence the way neurons integrate their inputs. And this project will unlock the key to understanding neuronal integration circuits. So I thank you to our host and everyone participating today. We very much look forward to the discussion and the exciting transformative work to come from this team and the community over the course of this project. Thank you.
Thank you, Corey. Thank you, Karen. And now with your blessing, we're ready to go. And Ellie, you can take us from here.
Hi, everyone. Welcome to the first session, circuit integration. And we'll get right to it to not be too much behind on time. Our first speaker today is Honghui Zhang from the Allen Institute for Brain Science. And Honghui will tell us about cell types and a connectivity in the mouse visual cortex. Please.
Okay. Good morning, everyone. Thanks, Rafa and Ellie, and Karen and Corey. I will start sharing now. Yeah, thanks for the great introductions. Okay, can you guys see my slide? Everything good? Okay, assuming everything is good. So today I'll tell you about the work we're doing at the Allen Institute in the mouse visual cortex. The visual cortex has been a major focus point for us over the past 10 years as a way to integrate our various research projects together. So today I will tell you about a really large body of work in investigating cell types and connectivity in the mouse visual cortex. I can only talk about this large body of work at a very high level. And my intent is not to tell you about a particular cell type, what it does, or a subcellular localization region such as the dendrite, but giving you an overview of the overall organization of cell types and the connectivities in the system and how they interact with each other. Hopefully, with this, my talk will set the stage for later today's talks, which will dive, I believe, a lot deeper into specific cell types and circuit function. So visual cortex is one of the major models in neuroscience for many, many years now to study circuit function. And here I just highlight a couple of studies showing the organization of the visual cortex. In the top, Andrea Burkhalter and Chuanxin Wang have used a detailed tracks tracing to delineate the anatomical organization of the visual cortex in the mouse. They found about 10 higher visual areas surrounding the primary visual cortex, and the extensive interconnections among those areas to form 2 major subnetworks for the mouse visual system, the dorsal stream and the ventral stream, or the medial and lateral stream, which are very consistent with what has been already found in the primate visual cortex, which also composed of 2 major streams, dorsal and ventral streams, which process the where and what kind of information about visual inputs. And also there's extensive knowledge about the many different cell types, especially neuronal cell types, excitatory and inhibitory, within the visual cortex, and their connections both locally among the excitatory and inhibitory neurons, as well as inter-area long-range connections between the different visual areas in this local environment, as well as with other cortical and subcortical regions. So this is really just a diagram from the primate showing the many connections that we already know about. Local connections in a vertical direction across the cortical column, as well as long-range inputs and outputs from the visual cortex into other parts. There's also a tremendous amount of knowledge already gained about the different kinds of neuronal types in the visual cortex. And here I just use one example from our own study in using patch clamp morphoelectrical characterization to identify all the major neuronal types, excitatory across different layers, as well as inhibitory neuron types, which was surprising to us to see that they are also organized very nicely in a layer-specific manner. So at the Allen Institute, we have been setting up high-throughput technology platforms to study cell types and connectivity in a comprehensive manner for a particular brain region or even for the entire brain. And here I just highlighted some of the major high-throughput technology platforms we have established and generated large-scale data sets as a foundational resource for the community. So very often our front-end approach is single-cell genomics approaches, and that includes single-cell RNA-seq transcriptomics, single-cell tag-seq epigenomics, and spatially resolved transcriptomics. From here, we gain essential and comprehensive information about molecularly defined cell types, and we use that information to develop genetic tools, including both transgenic mouse lines and now more in recent years, enhancer-based viral vectors to target specific cell types based on the information that we had gained from single-cell genomic studies. On the anatomical side, we use morphological reconstruction in both brain-wide mesoscale level using light microscopy, as well as a nanoscale level at synaptic level using electron microscopy to understand the morphologies, axon projections, and the connections among the different cell types. Finally, we also use a set of multimodal integrative approaches to link molecularly identified cell types with the anatomical and physiological properties of the cells. Approaches such as patch-seq, bar-seq, and anterograde retrograde tracing coupled with molecular profiling. Finally, to handle all these very large scale data sets, it is essential for us to establish computational pipelines that will allow the management of the data, analysis of the data, and extracting knowledge from the data. So with this general introduction of the approaches, today I will tell you about a series of studies starting from single-cell transcriptomics to understand the organization of the cell types in mouse visual cortex in particular. And as I said, I started with transcriptomics, and then I will move on to studies using patch-seq approaches, electron microscopy, long-range connectivity mapping, and finally linking everything together. So in the early days, we generated the first transcriptomic cell type atlas from the mouse visual cortex. And this is really the first ever transcriptomic taxonomy created in detail for a particular brain region, cortical region. For a particular cortical region. And just, so this is already really outdated, but it gives a initial view of how complex the cell type composition can be, even just from a single cortical area. And you can see here, this initial study, we only profiled about 1,700 cells by SmartSeq, and yet we identified about 50 neuronal and non-neuronal types in the visual cortex, and both glutamatergic excitatory neuron types and GABAergic inhibitory neuron types are organized according to the layers or depths of the cortex. So from here, we moved on to a larger study, and in this particular case, we analyzed 2 different cortical areas at the same time, with the question of trying to understand how how cell types differ or are common across different cortical areas. So in this study, we profiled about 24,000 cells, 10 times more than what we did before, in the primary visual cortex at the back of the cortical surface and anterolateral motor cortex at the front. And here we then again derived a much more detailed, comprehensive cell type taxonomy based on the transcriptomics. And the major finding from this study is that there exists a dichotomy between the two major classes of neurons in the cortex. The GABAergic inhibitory neurons are all common or shared between the two different cortical areas, whereas the glutamatergic excitatory neurons are highly distinct between the two different cortical areas. This is further supported by the much larger number of differentially expressed genes for each homologous paired cell type across the two different areas compared to GABAergic neurons. We think this dichotomy is probably due to the different developmental origins of those two major classes of neurons, with the excitatory neurons generated within the cortical plate itself, and thus subject to the morphogen induced gradient regulation and regionalization within the cortex. GABAergic inhibitory neurons, on the other hand, were originated in the subcortical ganglionic eminence areas and migrate into the cortex during development, and therefore maybe intrinsically they don't have this kind of region-specific information in their transcriptomes. Okay, then from here on, we further expanded our work, transcriptomic profiling, into covering the entire cortical surface. So that includes all the different regions, sensory, motor, and associational areas in the isocortex or neocortex, as well as all the different regions in the adjacent archaeocortical region, hippocampal formation. So overall, more than 30 individual cortical regions are profiled in this large-scale transcriptomic study, with now 1.2 million cells profiled by 10x droplet-based approach. So again, we computationally derived now a much larger transcriptomic taxonomy. We still see the same kind of hierarchical organization of the different cell types defined transcriptomically, with major classes, glutamatergic, GABAergic, and a number of non-neuronal classes. And below that, with subclasses and clusters or different types of neuronal and non-neuronal cells. The general organization is still very similar to what we have seen with the two cortical area study, with a lot of the cell types, GABAergic cell types shared across all the cortical areas, except for a small set of GABAergic cell types that are specific to hippocampus. On the glutamatergic side, on the other hand, there are many different types now, and many of them are region-specific, whereas many others are shared among neighboring regions and forming all kinds of gradient distributions, anterior, posterior, medial, lateral, and many continuous variations across different cortical areas. So this study reveals a highly complex cell type landscape across the entire cortex. So there has been a lot reported in this particular study. We mainly investigated the similarity or difference of the cell types between hippocampal formation and the neocortex, as well as the kind of a gradient distributions of cell types within the neocortex itself across different regions. And here, I just wanted to highlight one little piece of data that we have generated along in the study, showing the tremendous diversity and also continuous variation of a particular type of cells within cortical areas, and that is the intratelencephalic neuron types, or the cortical projecting neuron types that are distributed across all different layers of the cortex, from layer 2-3 to layer 4-5 to layer 5 and layer 6. So this large group of neuron types form actually a continuum, from layer 2-3 to layer 6. And their transcriptomic profiles correspond very nicely with their spatial localization across the cortical layers or cortical depths. So when you aggregate all the different IT neuron types together, the cortical boundaries become even fuzzy, and because of the tremendous continuous variations across the depths. And you know, as the audience here is interested in studying the cell types, excitatory and inhibitory cell types within a visual cortex, here I just wanted to use this to show the many different clusters that we have identified within the cortical regions. Some of them are distributed in different regions, but there is also a lot of shared types across regions. And the visual cortex, the IT neurons types within visual cortex are shown in this particular row here. And finally, after profiling the cell types across the entire cortex, we further moved on in recent years to profile cell types across the entire mouse brain. So this is yet another much larger scale study that we recently completed and will be published very soon, in which we used the 10x single-cell RNA-seq approach to profile more than 7 million cells across the entire mouse brain. And after a stringent QC criteria we have established, we have 4 million cells passing the QC. And using those 4 million single-cell transcriptomes, we computationally generated a very large cell type taxonomy now across the entire brain. And this taxonomy is again hierarchically organized into more than 5,300 clusters. And then they are grouped into supertypes, subclasses, and classes. We find extreme diversity, tremendous diversity, extraordinary diversity of neuronal types in this whole brain taxonomy, with 5,200 out of the 5,300 clusters are neuronal clusters. And they exhibit highly specific spatial distribution patterns, as shown by a companion whole brain scale spatial transcriptomics merged data that we have generated, which are not shown here. This is another way of showing the whole brain transcriptomic cell type taxonomy with 5,300 individual clusters, and the clusters, all 4 million cells profiled and used for classification, are shown in this UMAP. And the UMAP is colored by class, subclass, and region, and the neurotransmitter types. So a major take-home message for this particular slide is that we find that the cell types in the brain are defined not just by their molecular identities, for example, by the neurotransmitters they express, but they are also defined very strongly by region-specific signals. So in fact, the major classes that we have defined correlate very well with the anatomical regions where the cells come from. And it's really a combination of region and transcriptomic types or molecular identities. So, and we find a large set of transcription factors that define both molecular identities and regional specificities that collectively define the whole brain taxonomy. Okay, so what does this have to do with visual cortex? Here, I just wanted to use one slide to show that we also have dived deeper into specific brain regions to look at the neuronal cell type or organization within those regions, which we call a large neighborhood. And here in this particular slide, I show the major relevant neighborhoods are the pallium glutamatergic neighborhood and the subpallium GABA neighborhood. In the pallium glutamatergic neighborhood, we have all the excitatory neuronal types coming from the entire pallium organization, a developmental term which covers the entire surface of the telencephalon that includes all the cortical areas, not only cortex, hippocampal formation, but also the more ancient cortical areas such as olfactory areas, and the cortical subplate, namely the lateral basolateral amygdala areas. So we are able to, in this neighborhood, we are able to expand the understanding of the relationship between different excitatory neuron types that are very much region specific. From cortex hippocampal formation into cortical subplate and olfactory areas as well. We see this series of intrathalamus projecting neuron types, extrathalamus projecting neuron types, the layer 6 cortical thalamic projecting types, near projecting, and layer 6B subplate projecting types. This kind of major subclasses, categories of cell types that are expanded into all the different pallium or so-called cortical structures, being ancient cortical structures or more recent neocortical structures. So this general subclass level excitatory neuron organization is well conserved across all different cortical areas. Similarly, all the GABAergic neuron types within the telencephalon came from these subpallium structures during development, and they also have extensive homologous relationships with each other, and this includes all the cortical GABAergic neurons that were generated in subpallium, migrated into cortex, all different regions of a cortex, and we now can begin to better understand the relationships of the cortical GABAergic neurons and the subcortical GABAergic neurons within the telencephalon. Okay, so with that, again, without getting into details, I will now tell you about a newest data set that we have generated, which we are still in the process of analyzing, and that's we are preparing a manuscript. So this is a now shifting focus, focusing back onto the mouse visual cortex, but trying to understand the development of all the cell types within the mouse visual cortex. So this is yet another a substantial set of single cell transcriptomics data, including about 1.5 million cells profiled from the visual cortex, as well as neighboring cortical areas throughout the prenatal and postnatal development. As you can see here, this is a UMAP with all the cells grouped together, and the UMAP is colored by time points and by the major subclasses that we have been able to define from this data set. The time points covered from E11.5 all the way to P28, with everyday coverage all the way through P17. So this is a very densely temporally sampled data set. And from this, we are very much hoping that we will be able to generate unbiased trajectories of all the major neuronal types, major neuronal types throughout the development. And then we'll begin to be able to see when they emerge with time and what gene expression changes. As you can see here, there are dramatic gene expression changes from earlier time points to the adult stage time points. And we want to see what kind of gene expression changes are happening at exactly what time points. Okay, so with that, I want to move on to talk about then other kinds of studies. What do all those transcriptomic cell types mean? How are they related to the classically defined cortical cell types by morphology and physiology, and how they are connected to each other, and what their functions might be? So in this first multimodal integrative study using PatchSeq, we profiled nearly 4,000 individual visual cortical GABAergic inhibitory neurons, and obtained the local morphologies from 500 of those 4,000 neurons. So we have transcriptomic and electrical information for 4,000 neurons, and the triple modality information for the 500 neurons. So from this large data set, we examined the correspondence between the transcriptomic identities, T types of those cells, as well as the morpho-electrical identities, or the ME types that we have defined independently from the T type for those 500 cells. And you can see here the correspondence between T types and ME types are very high, but at the same time, the correspondence is not perfect. There's this fuzzy several to several correspondence, rather than precise one-to-one correspondence. And then we further integrated the transcriptomic and morpho-electrical information together to define the unified MET or triple modality types for the GABAergic neurons in visual cortex. And here are the 28 MET types that we have defined that are grouped into 5 major subclasses of GABAergic neurons. And they are very consistent with the existing knowledge of GABAergic neurons in the cortex. They include the parvalbumin positive types, somatostatin positive types that belong to the MGE branch, and also the LAMP5 positive neogliaform types, the SNCG positive multi-polar interneurons, as well as the VIP positive bipolar interneurons. So the overall conclusion from this part 6 study is that the MET types have intermediate granularity. There are only 28 MET types versus the 61 T types that we have identified for the GABAergic neurons. But the MET types have higher cross-modality concordance and mutual predictability. The MET types also recapitulate traditionally defined interneuron types very well, and with further subdivisions that are often driven by layer specificity. And
Kui, we have 5 minutes if you want to leave a few minutes for questions.
Okay, I will try. I still have a lot to talk about, but I will try to finish this quickly. Yeah, so the MET types, we think they can serve as a Rosetta Stone to bridge disparate molecular and morpho-electrical data together. So, and with that, then I will give you an example of how we use the patch-seq data as a Rosetta Stone to link transcriptomic cell types with cells identified from, for example, the electron microscopy connectomics dataset. And in this particular example, we identified a set of somatostatin positive interneurons in layer 5, and we know that in layer 5, layer 4, 5, they can be they are transcriptomically grouped into several different major types. There's the layer 4 targeting SST HPSE type. There's the layer 5 T-shaped layer 5 Martinelli cells, and also the fan-shaped layer 5 Martinelli cells. We are able to correspond the transcriptomic types with the cells in the EM data, and we find a very interesting preferential connectivity pattern among those 3 different types of somatostatin cells. The layer 4 somatostatin cells or Martinelli cells indeed heavily target the layer 4 excitatory neurons within the EM volume, and the T-shaped Martinelli cells, both T-shaped and fan-shaped Martinelli cells mainly target layer 2-3 excitatory neurons, but also in layer 5, the T-shaped mainly preferentially target the IT, the intratelencephalic neuron types, whereas the fan-shaped Martinelli cells primarily target the extratelencephalic projecting or pyramidal tract neurons in layer 5. So there is a differential connectivity, synaptic connectivity here for the different transcriptomic somatostatin types. So expanding that work into covering many different inhibitory neuron types within this EM column, which is 1 cubic millimeter covering the entire visual cortex cortical column. This is a matrix of inhibitory to inhibitory connections, and this is a matrix from inhibitory to excitatory connections. And very interestingly, what we find in this V1 column is the specific circuit motifs with different kinds of GABAergic interneuron types target very, very specific glutamatergic excitatory neuron types in different layers. So different individual inhibitory cells are involved, in the very, very specific local motifs. Almost none of them have this kind of promiscuous blanket coverage of inhibition of the cells, but they form very, very specific circuit motifs. Okay, I know I should end my talk now, so I should just very quickly, in 1 minute, also to talk about also the inter-area long-range connections that we have been studying. This is yet another level of work in using anterograde and retrograde tracing to generate connectivity matrices across different cell types and in different cortical areas. And the major finding from our major goal in looking at the intracortical as well as the cortical thalamic connections is to use the axon projection termination patterns to define the hierarchy across the different cortical areas. So we are able to derive this completely data-driven hierarchy both among the different cortical modules, as well as among the different primary and higher order visual areas. Using anterograde tracing long-range axon termination patterns. Similarly, at the same time, we also, in recent years, we used rabies-based retrograde transsynaptic tracing to define inter-area connections again, and we find strong reciprocal connections between the different cortical areas, and this is with a focus on the visual cortical areas, the primary and higher order visual areas. And we find that across the different layers, the input patterns to the different primary visual cortex is very similar. And we find a special feature for the layer 6 in visual cortex in that they receive much more stronger contralateral inputs. And that's focused on the deep layers. We also looked at the connections among the different higher order areas to understand the exact connection between the different streams, the dorsal streams and the ventral streams. We are able to confirm in a very strong differentiation between the dorsal stream and the ventral stream, and also the hierarchical organization of the different higher visual areas within those two different streams. And furthermore, in addition to the two major streams, we also find that there are further subdivisions within those two streams. For example, within the dorsal stream, we see two substreams, one on the lateral side and one on the medial side. And then secondly, between the dorsal and the ventral streams, we identified several different areas that are actually mediating, that are involved in both dorsal streams and ventral streams. And this includes the AL area and also the PM areas. So we find that these streams become a more of a topographic sub-network organization within the visual cortical system. And we just wanted to close that and now trying to link this kind of a long-range connectivity patterns to single cell level morphology and also to the transcriptomic types. And in our most recent work, which we just submitted to bioRxiv last week, again, we used the patch-seq data, now focusing on the excitatory neurons within the mouse visual cortex, using local morphologies, both dendritic morphologies and local axonal morphologies to classify the different excitatory neuron types, to correlate them with their transcriptomic types, and also correlate them with the long-range projection patterns of those excitatory neuron types. And we find a very good correlation that with high predictability across modalities. So with that, I just want to use this slide to summarize the work that I told you about today, and thanks a very large team of scientists at the Art Institute. It's really big science and team science that allowed us to generate this tremendous amount of data and knowledge and for the community. Okay, with that, stop sharing.
Thank you, Hongpei. So much data. I think that because the talks are relatively short and we have a half hour for a discussion at the end, we can take questions then. And to remind the attendees that you can submit questions through the chat for any one of the three morning speakers that we can bring up in the discussion. And our next speaker is George Keller from the Friedrich Miescher Institute. And he will be telling us about predictive processing, a circuit approach to psychosis.
Excellent. I hope you can see and hear. Thank you very much for the opportunity to speak. It's a pleasure to be here. So what I thought I would talk to you a bit about today is a story or an idea that we've come across that has completely changed the way I think about cortex, or has when we sort of first saw this about 2 years ago. Most of the lab has been focused on studying the computational role of the cell types. Hongpei has so beautifully elaborated previously, and just to sort of tie into this, I thought I would start on this point. We're not going to go into the details of these different circuit motifs, but that's sort of been what the lab has been studying, and the guiding principle that we've used is the computational idea of predictive processing. And we've been in essence, what we've been doing is putting, molecular tags or identifiers onto these different cell types that were proposed by the model, or in this toy circuit that Tom and I proposed a few years ago, for how this could work. Right, this is just one possible implementation of the idea of predictive processing in cortex. Anyway, I mean, and somewhat the beautiful thing that I love the work that Hongpei has just been talking about, and the EM data that came out of Nuno da Costa's team at the Allen and Hongpei's team on inhibitory neurons, right? I mean, it matches somewhat nicely onto this scheme. We think these triangles here should be pyramidal excitatory neurons, circles are inhibitory neurons. We think these are layer 2 or superficial in the mouse, superficial layer 2-3 neurons. And we postulated that they have a separate set of inhibitory neurons, which is exactly what sort of the EM data now reveals, that there is a separate set of inhibitory neurons that target layer 2 and deeper layer 3 neurons, or proto layer 3, if you. Anyway, I think this has potential sort of to go from these toy circuits to computational principles, but the idea that sort of got us away from working on this, that's sort of what I would like to talk to you about. And obviously it's related to this, and it was born in the idea of pursuing these computational ideas. And the basic question we were asking at the time, all related to predictive processing, is what is the significance of self-generated feedback? Basically, the age-old question. That's already what Hermann von Helmholtz asked in the 1850s, and often credited as one of the first to propose this idea, is that how does the brain know when an input is externally generated, and how do you know that something is the consequence of your own actions? Right? If you move your eyes, most of your visual input, I would say 99.999%, etc., of your visual input is actually self-generated. You move your eye every 100 milliseconds or so, you move your hands in front of your face, you move your head. All of this has direct visual consequence, and only a small minority of things that happen in your visual input are actually driven by external input. Right, so what is just to give you sort of the logic framework, or one derivation of this idea of predictive processing, there's a variety. Assume you're a brain with two areas. One, there's sensors, and on the other hand, there's effectors. There's just these two brain areas. Now, just conceptually, you might be interested in having a mapping from some desired sensory state on the left to the movements necessary to get there. Right? This can be relatively banal. My hand is over here, but I would like it to be over here on the left. To pick up something, it could be I'm hungry. What do I need to do to be not hungry? Or I would like to win the lottery. What do I need to do to that? Now, obviously, for some of these transitions, we know how to do this, or our brains, through experience with the world, have learned how to do this. We know these models exist. Sorry, this is jargon. They're called inverse models. That's what they were originally called in the 90s when this terminology was invented. But this mapping, we have this. I can give you some random Swiss German word that you've never heard before, and you'd be able to pronounce that reasonably well on first try without having to practice 100 or 1,000 times to get close via some random walk, right? On the first try, you're reasonably close. That's a relatively complicated mapping from an auditory stimulus to the control of your larynx that is necessary to actually reproduce that sound. And you have that inverse model. We know brains store this. The problem is this is relatively difficult to learn. A much easier model to learn is the sensory consequences of your own actions. And this has a simple reason that this is easier to learn, and that you have an easy teacher. And this teacher is just the thing we call the world, right? Whenever you act, that actually generates sensory feedback. And now you can learn this mapping relatively easily because you have this teacher that the actual sensory input. Anyway, this is the whole idea of predictive processing. Obviously, neuroscience has invented terminology for this over the decades. If you've heard of corollary discharge, this is sort of this idea before it's transformed into sensory space. Different copy is the signal. Oh, sorry, this is mislabeled. It's actually the other way around. Doesn't matter. One is before the form-model transform, the other one's after the transform. The point is that this signal can then be compared to sensory feedback, and this generates a learning signal, right? In predictive processing, this output is called the prediction, and this learning signal is called the sensory prediction error. But this is obviously sort of a blueprint for the way we think about reward prediction errors in the dopaminergic system, how we think about cerebellum. And I would argue, as we've sort of established, it has become clear over the last 10 years or 20 years or so, is also a reasonably good approximation for how cortex might work. Right. The way we investigate the type of experiments we can do, right, is then that's why virtual reality is useful, because you can relatively simply do this experiment where you decouple the feedback from the world. So what I'm going to show you, and sort of the question we started with, is what differentiates this condition, right? We'll call this closed loop, where the things a mouse does actually have consequences, from when it initiates a movement, basically that moment when it probes the world, when it does something, when there's no consequence. We'll call this open loop, just for the sake of argument. So there's a closed loop experiment when what the mouse does is coupled to what it sees, or an open loop experiment where the two are not coupled. What this looks like is, this is a closed loop environment where the mouse's locomotion is now coupled to this movement in this virtual tunnel. And I hope you can see this through the virtual zoom, but on the right, there's a mouse that can do whatever it likes, but has no control over the visual flow it's generating. It's just seeing a replay of a previously generated visual flow. Right, closed loop and open loop. And then the question is, how does this, how do these moments, how are they represented, or how they affect cortical activity? Technology we use in this particular experiment is wide field calcium imaging. What you should be seeing on screen is raw data of one of these recordings. If you're not familiar with the technique, what we're looking at is the entire dorsal cortex of the mouse. On the left, it's the mouse brain, and roughly indicate that the red area is what you should be seeing on the right. So visual cortex here is in the back, the orbitofrontal cortex is just at the frontal edge, auditory cortex here just visible on the edge, etc. So it's most of dorsal cortex that we can see with this technique. Now, the key is that we can do this in a cell type specific way. Now, I'm going to show you initially data from the two extremes, or from two of the experiments we did. One is where we used the PHP virus under a relatively ubiquitous promoter to express GCAMP in all cells. That's the example data I think you saw a minute ago, or on the previous slide. So I'm going to call this, for the purpose of talk, I'm going to call this brain-wide. It's not entirely accurate, but we can discuss the details if you're interested later. But it's most cells in the brain. And then we use a layer 5 TLX3 is just a Cree line that we can use to label layer 5 IT cells, a subset, or sorry, I should say a subset of layer 5 IT cells. Right, so how does this look? In this moment when the mouse starts a movement, right, it's in a closed loop. Sorry, the top row is data from closed loop. To read these plots, this is the entire, again, this field of view I showed you. Red means activation, blue would mean decrease in activity. Time 0 here after the second image is the running onset. And most of dorsal cortex responds in these brain-wide recordings when a mouse has a closed loop locomotion onset. This isn't a big surprise. This has been reported by many other labs that there's very strong movement-related activity, not just in visual cortex, but throughout dorsal cortex. Now, when you then look at the same movement onset now in open loop, there isn't too much of a difference. Now, you should, just to briefly pause, the thing we're coupling is locomotion to a part of the visual space, a relatively small part of the visual space. Everything else is unchanged, right? Auditory coupling is normal, somatosensory coupling is normal. Vestibulomotor coupling is in both cases equally 0, but the mouse is head fixed, so there should be very little vestibular input. But the only thing that changes between these two conditions is whether the visual input in a certain part of the visual space is coupled to locomotion. And the answer is, right, in most of cortex, you probably wouldn't have expected to see a big difference. In visual cortex, I would have expected to see a little bit of a difference. There is, if you look closely, you can see the difference. But the surprising thing, and why I'm showing you this, is that if you now look in layer 5, the situation is very different. This is one example mouse each. We'll show you average data in a second. The point is that in layer 5, across the entire surface, dorsal surface of cortex, you can tell whether the mouse should have movement onset with a small part of visual space coupled to its movement or not. Right? The difference, sorry, top and bottom is closed in open loop here now for the TLX3. You can from OFC, all the way at the other end of cortex in layer 5, decode whether the mouse had coupled movement onset in a small part of visual space, which is somewhat surprising to say the least. To sort of compress this and show you averages over mice, what we use is our favorite data compression method, which we call our favorite 12 brain areas, which is just these areas I'm showing you here at the top right. And then I can show you traces as a function of time of 6 areas. It doesn't really matter what the 6 are. Any 6 will do. These are just the ones we chose. Again, same data. Now just average across many different mice. Left at top, again, these open and closed loop running onsets of the brain-wide recording. And you can see it doesn't, there isn't much change. There are small differences if you look closely. There's very big differences independent of brain area. I mean, it is bigger in visual cortex, but the differences are visible, across all of dorsal cortex, independent of which brain area we look in. We can decode whether the mouse just had an open loop or a closed loop running onset. We can't do this from the brain-wide activity. So something seems to be very special about these layer 5 IT cells. They care a lot of whether the mouse is in a closed loop environment or in an open loop environment. And just to show you across many different mouse lines, we haven't only done this for the 2 I'm showing you. We actually explored this a bit further. And that the data I'm showing you on the left is this black 6 data. This is the brain-wide recording. TLX3 is this layer 5. EMX1 is all cortical excitatory neurons. COX2 is a layer 2-3 line that labels an undefined subset of layer 2-3 excitatory cells. SCAN1A is a layer 4, similar for layer 4. It's a subset of layer 4 cells. NTSR1 is a layer 6 line PVS. The IPNS should be clear. Right. So the point is there's something special. Sorry, and what we're quantifying on the x-axis here is how similar on average these closed and open loop traces are. Just this 6-second or so window of time around the running onset, how similar is that? And the lower the number, the more dissimilar these 2 activity patterns are on average. For most cell types, they seem to match quite well. For layer 5, it seems to really care about whether there's closed or open loop. We don't fully understand what this means or why this happens, but there's something peculiar about these deep layer cells. Right. Now to explain the next bit of data, which is again something we don't fully understand, but has, I think, interesting ramifications. And that's why I want to talk to you about it. So we were pursuing this question, what does it mean when something is internally or externally driven, as I tried to explain at the beginning. We have a series of compounds that we don't understand how they work. but we know they ameliorate this distinction in patients that have difficulty segregating. Right? And what I'm talking about is psychosis and antipsychotics. So in patients that hallucinate or have delusions, we know there's a list of compounds we can give them that we call antipsychotic drugs because they're efficacious against psychosis that prevent people from hallucinating and reduce their delusions. Obviously, we know a lot about which receptors they bind to, et cetera, but we know very little about what they do in the circuit. Right? How do they affect this circuit property? And obviously, what we then thought is we would give these mice, now that we've identified the cell type that actually seems to care a lot about whether the mouse has control over the environment or not, we give a mouse these antipsychotic compounds and see, is there a cell type specific effect? That was the logic. Right, of the experiment. Here's the data I showed you previously. Again, the open and closed loop running onset activity traces in brain-wide and these layer 5 cells. This is before, obviously, in the naive mouse, no antipsychotic. But then you can give the mouse antipsychotic and come back 2 hours, 24, 48 hours later, and we look at the activity. Now, what you should see at the right here, top right, is the same activity in the brain-wide recordings after clozapine treatment. Clozapine is one of the examples of the drugs I'm going to show you. We've done this for a variety of different antipsychotics. This is just one of the examples. It's a frequently used antipsychotic, frequently used in schizophrenia patients, of course. Right. And the point I'm trying to make is here, there isn't all that much of a difference. The inset compares the two traces for V1. You can see differences. But they're relatively small. And the relevance of this is, keep in mind, this is the experiment that we can do in humans. Right? Brain-wide recordings are, using either fMRI or EEG is what we can do in humans and have been done, thousands of times over the last 100 years or so, or 70 years since the discovery of the first antipsychotics, to try to understand which brain regions are affected by antipsychotic drugs. Now, if you just look at these layer 5 IT cells, the situation is completely different. You see humongous differences, right? Please note that the scale, that these responses now, these movement onsets are suddenly 5 to 6 times larger. They flip sign. Again, the inset is the comparison for V1. In the case of V1, this flips sign even. They become more similar. We don't understand why any of this is happening, but there seems to be a very selective effect of the antipsychotic drugs on the same cell type that seems to really care about whether the mouse is enclosed or open loop movement onset. Just to illustrate how large this effect is, I can demonstrate the size of the effect by using raw data. We don't even need to average. This is a brain-wide recording of before and after clozapine, just 10 seconds of recording or so. And I hope this transmits on Zoom with the video, but the point here, there isn't much difference. Again, if you know what to look for and if you look closely, you can see differences or effects of the antipsychotic. If you look in layer 5, the differences are much, much larger. And again, I hope you can see this. Otherwise, please, you'll have to believe the data I showed you before. But there's relatively strong single trial drastic effects of the antipsychotic just on this layer 5 cell type. Right, so we've learned a little bit about this, and I'll try to end on this. In a nutshell, I'll show you the data or try to convince you of this. But the point is that what we think these antipsychotics relatively generally do is they decorrelate the activity, the long-range activity. So they reduce the correlation of, layer 5 V1 with layer 5 ACC across all pairwise interactions. Oops. Across all pairwise interactions. To quantify this, we just take 5-10 minutes or so of recording and compute the correlation between different brain areas using this wide field method I showed you earlier. And this gives you these correlation matrices. We can do this before and after clozapine, and you can see this matrix on the right is a bit bluer, means the correlations drop a bit. The effect is much larger if you look at this in layer 5. The way we quantify this is, again, plot this correlation matrix now as a function of distance. So every point, every square in this matrix becomes 1 point in this plot, where the x-axis is the distance between the two cortical areas. And you see that the the larger the distance between the two areas, the lower the correlation. This isn't a surprise. We represent the data with an interpolated heat map, just so it's easier to visualize. I'll show you a bunch of these heat maps, but this heat map, again, y-axis is correlation, how similar the two areas are, x is how far apart they are. Right? So this is the data I showed you before. This is the black 6, so brain-wide recordings before and after clozapine. The fact that this cloud is on the right is a bit lower than it is on the left means all correlations have dropped. The white lines are just to guide your vision. This is just a copy from the left, a sort of ISO contour line from the left plot. If we look in layer 5, this is what this looks now in this representation. There's a very drastic effect on these TLX3 neurons. Correlations drop by almost a factor of 2. And now here's the totally bizarre thing that we found is if you look in layer 2-3, there's a COX-2 line now, it's a subset of layer 2-3 excitatory neurons. We barely see any effect. Right. This sort of makes sense that black 6 is a mixture of all kinds of cells, including layer 5 cells. So we expect, to see some intermediate state in the brain-wide recordings, but there's very little decorrelation in layer 2-3. So this is one of the sort of flabbergasting, if you will, findings that we've had. Just to show you this, sorry, that's my timer. I'm almost done. Just to show you this is consistent across antipsychotics. Here's 2 more. In the meantime, we have another 4 that all exhibit the same effect. They decorrelate layer 5 activities. This is aripiprazole and haloperidol, which are, chosen to be representatives of the different generations of antipsychotics, to be as far apart as possible in sort of the drug space of antipsychotics. And all of them, despite having very different receptor binding profiles, have very similar functional effects on these layer 5 cells. All right, so I hope sort of in summary to have convinced you that very simply what we think they do, these layer antipsychotics, is reduce the lateral correlation between all pairwise, all interactions in cortex selectively in layer 5. Right? And sort of just as a very hand wave, if you permit me, sort of a justification of why this might make sense, right? If layer 5, we think predictive processing postulates that layer 5 has something to do with the conscious perception. Assuming that this is the case, and assuming these layer 5 correlations are decreased by antipsychotics, this might explain why they work. Assuming that, if your layer 5 auditory cortex, let's say, is active, then you might hear voices when you shouldn't, right? Some spurious activity in auditory cortex or fusiform word form area, or somewhere in the auditory system is too high. That might lead to the conscious perception of speech, auditory hallucination. Now, if you had a way to decrease all lateral communication in layer 5, that might systematically reduce the chance of spurious activity in those layer 5 cells. So I don't think this directly relates, or probably doesn't directly relate to the disease itself, but it might explain why antipsychotics work. More generally, right? Now, what are we doing right now? We've completely switched sort of our research from the circuit elements of predictive processing to investigating this effect, and are seeing, right, there's 80 or so FDA-approved antipsychotic compounds. Is this common to all of them? We've only tested 8 so far, and we don't know, right? This is ongoing work. And the other thing that we're really interested in doing, and as Hongkui has already introduced, we're making an answer and promoter viruses that allow us to target these cells in both mice and non-human primates. All right. So what's sort of the problem? What's the big issue with this? And I'll end on this. Is we have this idea of brain function, as I've tried to explain. We have an idea of how this might map onto cortical circuits. The issue with what we found is that we have a very strong decorrelation effect in layer 5, very little effect in 2-3. Now, most models of cortical function, right, whether it's one from a textbook or whether it's this toy model for predictive processing, all the models I know have a very strong vertical interaction within the column of cortex. So thalamic input goes into layer 4, is related to 3, and from there to 5, right, which would mean there's a very strong vertical communication. And the computational unit is the column, right? This is what people have assumed for a long time, including myself, until we saw these data, right? Now, this is very hard to square with a circuit that you can expose to a compound that does very little to 2,3 and has massive effects on layer 5, right? And now, obviously, this is an antipsychotic. You have to know that aside from a 2-hour window of sedation, human subjects, non-schizophrenia patients, have very little effect of these antipsychotics. They don't report feeling anything, right? Obviously, this is mice, and the self-report is in humans, but also behaviorally, we find no effect on these mice 24 hours later. And these effects of decorrelation, they last up to 2 weeks. They're from a single dose of antipsychotic. It's very long-lasting. Doesn't seem to have any behavioral effects on the mice. So how is this possible? And this idea that they might, this points to the fact, which is what I was getting at, is that maybe superficial cortical layers 2, 3, and 4 are a separate computational unit from 5, 5, and 6. And by separate, sorry, I'm basically done. Separate computational unit, I mean similar to what we think about cortex and striatum being separate, right? Obviously connected, obviously very relevant communication. We're not the only ones who see this. Botan Droschka, a colleague here in Basel, recently had a paper where he showed that anesthesia also has a very selective effect on layer 5, which is equally difficult to explain. And recent work from Josh Wang's lab and Churchill's lab also find very different activity patterns in superficial and deep layers. So all this is consistent with this idea of 2 pathways, 2 computational units. Obviously, also the thalamic input. I forgot to mention work from Kevin Martin, Rodney Douglas, and Randy Bruno later showed that there's these 2 parallel pathways into deep and superficial layers. This is exciting. I'm excited about this because there's machine learning architectures that rely on this joint embedding of 2 networks. And this, while I have nothing intelligent to say on why this might be relevant, in machine learning, it's a very powerful concept that was recently developed, this joint embedding that more or less solves the problem of needing thousands and thousands of training examples. Not sure it's understood why this works in machine learning, I mean, but this is why I think this idea of separate networks might be relevant for us too. Thank you very much. That's it.
Thank you very much. Anyone who has questions for George, please put them in the chat and we'll come back to them in our panel discussion later. And we will move on to our final speaker for this session, Jerry Chen from Boston University, who will talk about functional and transcriptional dissection of cortical circuits during behavior.
Hello, everyone. Hopefully you can hear me and hopefully you can see my screen okay.
Yep.
Just start the slideshow. Okay, great. Well, thanks very much for the opportunity to talk about the work in our lab and how it potentially relates to the combined grant program that your team has put together and is kind of moving forward on. So thought I'd talk about a story that I think kind of relates to some of this research areas, both connecting the work from Han Kui as well as Georg. And so our lab is interested in understanding the circuits that are related to both perception and learning as it relates to behavior. It's a very long-standing question in the field, and we're approaching this question using the mouse sensory motor system, the whisker system. So it's a little bit different from the visual system, but I think also relevant for the mouse. It's a very highly neuroethologically relevant system for the mouse. Mice feel the environment with their whiskers, and you can study cortical circuit processing and primary somatosensory area, the barocortex, and how information is being processed from individual whiskers to these baro columns. And in thinking about perception and learning and how it relates to the program here, I thought I would start with just kind of thinking about a single neuron and how it's able to potentially carry out aspects of perception. So this is a review taken from Matthew Larkham, in which he's kind of proposes the idea that perception arises from interactions, right, from the external environment as processing the world around us, but then you're also and trying to interpret these physical stimuli using either internal models, predictions, as Georg kind of mentions, that we've learned through prior experience. And cortical pyramidal neurons are organized in morphology in a way that potentially allows those neurons to integrate and tease apart these different types of information. So presumably, basal dendrites of pyramidal neurons are receiving, kind of bottom-up external information from the world, either from layer 4 or layer 5, 6 from the thalamocortical areas. And at the same time, these pyramidal neurons are extending apical dendrites up into the upper layers, layer 2, 3, layer 1, where it's presumably receiving feedback connections, right, that may be conveying, internal models or predictions. And so these two compartments of the neuron potentially has ways to integrate and combine this information and interpret, the world around us. And so the idea is, how do we think about circuits that might be able to kind of, work on these internal single neurons to potentially carry out these computations? And so there kind of leads to the idea of cell types, which Han-Kui has covered quite extensively. And we can think about cell types in a number of ways, either in terms of morphology, their connectivity patterns, their physiological properties. Many of these types of properties are defined on a molecular level through gene expression patterns that start to potentially dictate these kind of anatomical and functional properties. And so it's been a large effort to try to understand, are these cell types organized into potential what are called motifs that might be able to carry out these kind of elemental types of processing or computations that underlie these aspects of either learning and perception. And so if you start to build up this, going from a single neuron where you have excitatory neurons, where you have these apical inputs and these basal inputs, one potential motif that's been discovered involving these inhibitory VIP neurons and inhibitory SST neurons provides a way for some of this information to be gated and for this information to be graded. So the idea is that VIP inhibitory neurons are receiving potential top-down inputs from layer 1, and these, when they're driven by these top-down inputs, then they would inhibit somatostatin inhibitory neurons. And these somatostatin inhibitory neurons then target the apical dendrites of local pyramidal neurons, such that when there's top-down inputs coming in, this allows for this disinhibition that enables this top-down input to be integrated with incoming sensory information. So this type of motif has been identified and observed across multiple cortical areas, and the question is, are there other motifs like this that exist, and what types of computations could they potentially perform? And so, as Han-Kui highlighted, there's been a large work carried out by her team and others to map out the numbers of cell types there are in the brain. And according to this study, where they mapped out the cell types in isocortex, they identified on the order about 300 cell types, which can be identified by some combination of transcriptional gene expression patterns. And so this really opens up the question in terms of whether or not these particular putative molecular cell types have unique functional properties. So in order to understand the in vivo function of these particular cell types, traditional methods in order to identify cell types either have been, in a mouse at least, have occurred through transgenic labeling strategies, or maybe post-hoc immunohistochemical labeling. And these approaches, while revealing, are also limited in some ways, in the sense that on one hand, you can really only interrogate one cell type at a time in a given animal. And two, a lot of these markers are labeling one gene. A lot of these cell types are actually identified with combination patterns of genes. And so this really kind of scratches the surface in terms of the gene expression necessary to really kind of dive deeper into specific subclasses and subtypes of cortical neurons. So along the way, there's also been a lot of technology development in the area of spatial transcriptomics, which allows you to multiplex and interrogate gene expression patterns in situ at high numbers of genes and differentially expressed to really kind of pull out cell type information. And so our lab worked to develop a platform that we call CRAC, which stands for Comprehensive Readout of Activity and Subtype Markers. And I'll show you this animation for how it works. So our lab uses 2-photon calcium imaging to monitor population activity in vivo as animals carrying out a given set of tasks. And at the end of our behavior sessions, we can take the brain out and we can section the brain, and we can find the same neurons that we'd image in vivo as we did in our ex vivo sections. And then we can take this brain tissue and then subject it to multiple rounds of HCR fish. And the HCR fish has a really high signal to noise, and also using spinning disk microscopes gives us really single molecule resolution, where you can start to detect individual mRNA transcripts and start to quantify their abundance in a given set of cells. And the kind of real power of this approach is that we can multiplex, so we can stain for a certain set of genes, we can strip and stain for a different set of genes and reimage, and across multiple rounds of staining and imaging, then you can start to build up gene expression patterns that will allow you to identify with more detail the exact cell type that you've been imaging, and start to correlate the activity patterns with their molecular profile. So using this approach, we started to look at the layer 2-3 cell types in barrel cortex. So we use on the order about 16 genes that labeled 3 major excitatory neuron classes, ADAM-TS2, BAS1A, and ASMAT. And then inhibitory neurons, the major 4 inhibitory classes that we can also further subdivide into subclasses of inhibitory neurons. And kind of long story short, for one of the findings that we discovered in our study is that one of the cell types that we've studied, this BAS1A cell type, is something that we believe is involved in as operating as a circuit hub in sensory processing. And I'll kind of walk you through the evidence that supports this information here. So again, we train our animals to a whisker-based task here. So animals are performing this whisker-based delayed non-matched sample task. As we perform population imaging, we can then use generalized linear models to decode the information that's encoded within each individual neurons by regressing the activity to different task variables. And then we can start to study how these encoding of task information differs between different sets of cell types. And here I'm just showing you the results from the 3 excitatory layer 2-3 neurons that we compared. And one of the first things that we kind of saw readily was that if you just look at the average firing rates between these ADAM-TS2, BAS1A, and ASMAT neurons, that BAS1A neurons tend to have overall higher firing rates compared to the other 2 cell types. The next thing we looked at was the degree in which these cell types were encoding task information, either whisker kinematics or the direction of the rotor that we are deflecting the whiskers. And overall, the BAS1A neurons show higher encoding of these whisker-related features. Now, you might think that maybe this higher encoding might be due to the fact that these neurons are more active. If we look at other task variables like whisker touch, if you just deflect the whiskers, for example, we don't see any differences in how the different neurons are responding to just whisker deflection. It really seems that these BAS1A neurons are really operating as some sort of kind of highly selective feature detective compared to the other 2 sets of neurons. The other thing that we can do is, because we're monitoring population activity and we were able to identify the neurons, the cell types within this population, is we can just look at the functional connectivity patterns between these different cell types. So what we did was we trained a generalized linear model on one neuron and then ask how well could that neuron's activity be explained by the different task variables. But then we added in the activity patterns of all the other cell types that were measured simultaneously. And from this, we can generate these network graphs that represent some functional connectivity between a given set of neuron and its other image cell types. We can generate these network connectivity graphs across controlling for different task parameters. And what we find in general is that BAS1A neurons, their activity was highly explanatory of other neurons within the population, suggesting that they're potentially an orchestrator of local network activity. In contrast, if you look at somatostatin neurons, these neurons, their activity can be explained by the activity of the surrounding population, suggesting that these are a follower of local activity patterns. We can then look at the strengths of the functional connectivity between any given pairs of cell types, controlling for different task variables. And what we found consistently that was a strong functional connectivity between somatostatin neurons and the BAS1A neurons, suggesting that potentially there was some sort of anatomical connectivity between these two cell types. So to kind of address this, we turned to monosynaptic transsynaptic rabies tracing. So we took two sets of transgenic mice expressing Cre and somatostatin neurons, and then as a control, mice expressing Cre and VIP inhibitory neurons, and we infected these cells, cell types using AAVs that expresses the rabies receptor. So serving as starter neurons that we can go in with rabies later to infect these neurons and map the inputs of either somatostatin neurons or VIP neurons. And then for every green cell that you see here, that we subjected them to our HCR fish and asked what type of cell, what are the cell type of these input neurons to these somatostatin or VIP neurons. And what we found was there was a higher degree of connectivity from BAS1A neurons onto local somatostatin neurons compared to VIP neurons. This confirms the idea that these BAS1A neurons, which are function coupled with somatostatin neurons, there's an underlying anatomical substrate to this functional connectivity. So this puts together a certain picture that we propose in terms of a cortical motif that we think acts kind of complementary to the VIP somatostatin disinhibitory circuit. So the idea is that if there's strong sensory input coming into the system, this will drive the BAS1A neurons, and then these BAS1A neurons, since they're connected to somatostatin neurons, will then inhibit the apical dendrites of the local pyramidal neurons. This is in contrast to the disinhibitory circuit that you see with the VIP neurons. And this potentially acts as a way for the local pyramidal neuron to be able to toggle or selectively integrate different sets of inputs depending on different sensory conditions. So for example, if there's a lot of salient information coming into environment, then that will readily activate the bottom-up circuit, shut down the apical dendrites, and allow integration to occur between the local lateral connections. In the case in which maybe sensory information might be weak, or there might be some differences between what we're receiving versus what the feedback is providing, then the top-down circuits are then activated, or more robustly ungating the apical inputs, allowing a further integration of the top-down and bottom-up information. Okay. So that summarizes one half of what we found. The other half that we found was kind of focused on the idea of plasticity and how these neurons potentially might respond in the cases of changing sensory experience. And the idea is that the genes that are identifying these particular cell types, some are presumably static, but others are highly dynamic and changing the experience. Immediate early genes, for example, are those that are underlying neuroplasticity. And one of the things that we noticed when we looked at the gene expression patterns of the BAS1A cells was that they were highly enriched in some subsets of immediate early genes such as FOS or NPX2. And this led us to wonder whether or not this enrichment of immediate early gene expression was a dynamic or stable property of the cell type. So what we did was we looked and using FOS-GFP as a marker of FOS expression, of immediate early gene expression. We took these animals and then we performed a sensory deprivation experiments where we trimmed all the whiskers except for a small little whisker that we could deflect. And this deprivation experiment has been shown to drive plasticity in sensory areas in the whisker cortex. And so what we did was we followed the FOS dynamics, FOS GFP dynamics in response to sensory deprivation. We can also monitor the activity of the neurons as well using our RCamp expression. And then after this experiment was finished, we then identified the cell types from the neurons that we were imaging in response to this deprivation. So first, if we just look at the FOS GFP expression, kind of ignoring the cell types, on a functional level from the RCamp, when you trim all the whiskers, you find that the activity of in S1 goes down within about 24 hours, but then you see a slight homeostatic recovery of responses. This has been demonstrated by Dan Feldman and others. If you look at the FOS GFP expression, what we find is that there are three general classes of FOS GFP neurons. One's where there's just stable low expression. Other neurons where you see dynamic expression, where GFP goes up or down. And then also neurons occasionally which are stable, which is consistently showed strong GFP expression. And overall, within the first 24 hours of deprivation, the responses of all three types of neurons goes down. But then within 5 days, so what you see is that there's a homeostatic response to the neurons that are highly and stably expressing FOS or FOS GFP. Now, if we kind of look at how this FOS GFP expression relates to the cell types, what we find is that if we look at the number of stable high expressing cell types before deprivation across the three excitatory cell types, what we find is that overall the amount of stable high FOS GFP expression is pretty consistent across the three cell types. But then after the bilateral whisker deprivation, though, only the BAS1A neurons maintain this kind of robust FOS GFP expression. The other ones are more dynamic and responsive to this deprivation. And similarly, then, if you look at the functional responses between these cell types, what we find is that within the first 24 hours, activity goes down across the three cell types. But then after 5 days of binocular deprivation, it's these BAS1A neurons that show this homeostatic recovery. So this suggests to us that not only are these BAS1A neurons operating as some sort of kind of maybe sensory hub for feature detector, they're also somehow maintaining potentially some sort of sensory stabilizing or maintaining sensory representations in the face of potentially new, novel, or changing experiences. And just to kind of bring this back to visual cortex, which I think is a big theme of this particular of grant mechanisms. I think since our work, I think Georg Keller has also discovered that these BAS1A neurons, which are also called RAD neurons in visual cortex, showed a strong are strongly driven by sensory neurons. At the time, we didn't have we couldn't figure out what the ADAM-TS2 neurons were doing in S1, but in his studies, interestingly, he shows that these ADAM-TS2 neurons are potentially these sensory prediction neurons, these mismatch neurons. And also a recent work from Larry Sibersky has shown that within layer 2-3, that the specification of these layer 2-3 cell types are very much driven during postnatal development after eye opening, and that dark rearing or sensory deprivation also changes the functional properties and also the molecular compositions of these neurons. So I think something to kind of chew on maybe, and maybe as a point of further discussion for later. Okay, so with that, I just want to conclude then by that we in a whisker system, we use both task behavior and sensory deprivation, subjected animals, imaged in during these different paradigms to monitor activity, and combine that with post hoc translational profiling. And through that, we identified, a particular type of cell within S1, and that could be related to cells in visual cortex or have unique roles in sensory processing. And with that, I'll just thank the people that carried out the work. So all this work was carried out by Cameron Condolis, a PhD student in the lab, and close collaboration with Basilica and Han Kui and at the Allen Institute. And with that, I'll happy to take questions.
Thank you, Jerry. Great timing. So maybe we'll roll the questions into start the panel discussion. And there are a few questions in the chat for Honghui and York, and maybe we'll come back to you right after that to wrap up. So Honghui, did you want to take the questions from Felipe?
Yeah, I think the question is about the continuous distribution of IT neurons across the cortical depths in the visual cortex rather than in hippocampal formation. So we don't know yet. I mean, this is just our observation. The cells form very fine layers or sublayers, and cells with and in different sublayers have distinct transcriptomic profiles, although it's a continuous distribution. So there's really fine-grained transcriptomic changes across the depths. We don't know about the functional implications yet, but just I think the deeper we look, the more specificity we'll find. Just like as Georg was talking about, and we find so many fine-grained specificity, for example, in layer 5, not only the traditional definition of 5A with IT and 5B with ET neurons, but we also find like upper, lower, and things like that. And based on our EM data, I think that this kind of fine-grained layer specificity probably will correlate with some kind of a connectivity, local connectivity specificity. We now have also begun to see that in layer 2-3 as well. Upper versus lower layer 2-3, or maybe even middle layer 2-3. Of course, different types of cells would have, they don't have the absolute sublayer specificity. It's just kind of a gradient, the preference in the upper or lower part. But that kind of a difference is often associated with connectivity specificity, either locally or with long-range connections, nearby areas. Another example is that Ed Callaway showed that the V1 neurons project into the medial part of the higher visual areas versus lateral part of the higher visual areas. They are also have the kind of a differential depth distribution in layer 2-3. So I think that we'll discover more and more specificity across the depths of the cortex.
So I was actually wondering, also taking it to what Jerry just showed, when he has this very specific circuit motif where the BAS1 neurons, have a specific role within that circuit, and you show so many different flavors within that group of L23 neurons. And I'm wondering, Jerry, how you're thinking about that, where does that mean that, the organization of these modules is maybe not so, like the cortical circuit module idea might not be so generalizable, but that there are these modules that are actually much more function specific, which is somewhat which is being kind of reflected in the having so many different subtypes within the same laminar region. I don't know if that question was clear enough, but do you understand what I'm asking? Jerry?
Yeah, I mean, on my end, I think, the motif that I kind of displayed was definitely a very gross generalization of probably one of many types of motifs that might exist. But I think that, and it could be there to carry out specific functions in a feature, as you say, in a very feature specific way, right? And I think there is some component, at least the way I'm thinking about some of these things in terms of, which compartments, along certain sets of neurons, might these motifs be targeting, that they might be influencing the integration of information in different ways. Also, then also to think a little bit more broadly in terms of long range versus input and output, right? There might be certain things that are happening locally, but then that information needs to get sent to different parts of the brain. Presumably, maybe these different gradients, these different areas are then allowing for that information then to be routed, in different ways.
Yeah, I guess, in the way that Honghui does the data analysis, I wonder if you have these sort of specificity module kind of intermixed with each other within the same region, right? Because they're looking at or implementing different features. That's sort of being reflected in what Honghui is actually seeing.
Yeah, yeah. Can I quickly address that?
Yeah.
Your comment question. Yes, transcriptomic profiles are actually very high dimensional, right? Because they encode for all kinds of information, including physiology, connectivity, and they may not be directly all completely correlated. So we and that's why there's all this continuous variations, and you can group cells into different clusters. Recently, in the most recent bioRxiv paper that we just posted, Stacy Sorensen, Nathan Gowens, and others at the Institute, they've done a detailed analysis of the patch-seq data of the excitatory neurons that you talked about, layer 2, 3, IT, ET, things like that. So they identified multiple principal components within the transcriptomic profiles, right? PC1, 2, 3, 4, 5, things like that. Usually PC1 and sometimes PC2 were correlated very well with clustering because it used the major variables, right? Variations within the data to group cells into types. But when they look at the correspondence of specific electrophysiological properties or morphological properties, they find that some of the physiological morphological properties correlate PC1, but often the properties could correlate with PC2, PC3, PC4. Things like that. So you can find transcriptomic correlates of specific physiological properties, but it may not be reflected at the cluster level because clusters, you cannot use all PCs to cluster because then it becomes too many tiny clusters. But yeah, so that's a very good example to illustrate the complexity that you mentioned.
It could be there are just a lot of really tiny clusters.
Yeah, well,
biologically. Yeah. Rafa, did you want to ask your questions directly?
Sure. Here's a question for Honghui. So your definition of a glutamatergic cell type, you said, well, it's both the transcriptomic profile and the location where it is. But at the end, the transcriptomic profile is determined by the transcription factors expressed at particular location. So I guess what I think you imply, what you're trying to say is that it's all about the transcription factor code that defines the cell type, and because different positions along the axis of the developing nervous system have different combinations of transcription factors responding to different gradients, that's where you get the cell type. So that wouldn't that argue that if you want to catch what the answer to the question, what is a cell type, is the transcription factor code?
Yeah, that's a great question. Really excellent question. I've been thinking about that as well. It is all about transcription factor code, exactly. And so the transcription factor, as I said, the transcription factors could define identities, and another set of transcription factors could define locations. But then you are saying that maybe the identities are also related to locations. So in the end, everything is position driven. In the adult stage, currently we have a counter argument against that. But maybe when we go back to the development, you are still right. But let me just talk about the current counter argument. So within a particular location or position, for example, there's always the mixture of GABAergic neurons and glutamatergic neurons. So even though they are exactly in the same location, they have completely different transcriptomic profiles and cell types. And they are driven by different transcription factors, distinguishing GABAergic from glutamatergic, right? Even though they are exactly in the same location. So that's one example against it. Another example is like, for example, layer 5 IT and layer 5 ET cells. They can also be commingled in the region, but IT cells and ET cells diverge early on during development, and they are driven by very different set of transcription factors. So even though ultimately they are in the same spatial location, they are driven by very different transcription factors. And then IT neurons, of course, across different layers are driven by different transcription.
Yeah, but wait, I think for the interneuron case, they're born somewhere else. So the transcription, the location that matters is where they're born, not where they end up.
Well, in the cortex, that's the case. But if you look at subcortical regions, for example, hypothalamus or ventral pallidum, the GABAergic neurons and glutamatergic neurons are intermingled, but they are still segregated completely.
I think, the prediction would be that, for instance, the IT cells would differentiate, will have different, they would originate at different times in development. I don't know.
They originate at different times.
Okay, so that would also agree
and maybe even different progenitor populations, or the same progenitor populations that are changing with time. Exactly.
That's right. So that's still consistent with transcription factor code idea, which, by the way, is not my idea. I credit Sidney Brenner. He's the one who came up with this in my book, and supposedly it can apply throughout the rest of the body, not just to the brain. So why would the brain be any different? So it's just about be at the right place at the right time. You have a particular type of transcription factor, you become something, and then if you mingle and do business with other people in your future, that's up to you. But you still remain with your accent, so to speak, where you're born. Thank you.
Yeah, exactly. Transcription factors specify the spatial temporal code. Exactly.
So there's a couple of questions in the chat for Jörg from Hiro Morishita. So the first question is, do antipsychotics also impact interhemispheres correlation?
Yes, they do. But we've only been able to establish a relationship between the distance between cortical areas. It's not entirely clear whether there's something special about interhemispheric connections. They tend to be longer, but even within a cortical area, we see the same relationship. So it seems to be distance and not necessarily a hemisphere that determines the primary effect. We can't rule out the hemispheric contribution with the data we have currently.
Although layer 2-3 neurons also have long range interhemispheric connections, right?
They're very important, just to be clear on this. We have no idea what's driving these functional correlations. It might be direct connections between the neurons. It might be subcortical loops. But this is exactly what we're trying to understand at the moment, is what is the contributing factor to these changes in correlation. And we don't know. It might be direct or indirect. And yes, of course, it's selective to layer 5, even though as you point out, layer 2-3 also connect, but those connections don't seem to be effective.
So it's not specifically the interhemispheric aspect of it.
Correct. Yeah, currently with the data we have, distance is good enough to characterize the effect.
And the second question from Hiro is, it appears the effect of drugs on layer 5 neurons were weak in ACC compared to other cortical areas. Is ACC different from other areas such as V1?
To be honest, I'm not entirely sure what this is referring to, but I think, I just had a quick look again at the data. To me, it doesn't look like they're much weaker or much different ACC, but this is a nice sort of point. Whenever one looks at these wide field imaging data sets, there's a very, very strong hemodynamic artifact. So there's always a contrast one needs to be aware of. We try to contrast different mouse lines against each other and hope that the hemodynamic occlusion is relatively similar in the two mouse lines that we contrast. ACC has this problem that it's very, very close to the midline, where there's relatively large hemodynamic occlusion effects. They come about simply by blood vessels shrinking and expanding in a brain activity dependent way. to the point where we get beautiful hemodynamic GFP signals. So if you do this wide field with GFP, you also see these signals. Anyway, this hinders us in comparing effects in ACC to more lateral, or RSC for that matter, to V1. Anything close to the midline is very strong contamination. So it's a bit tricky to answer. But as far as we can tell right now, there isn't any specific brain area that's particularly affected. And I assume this is in reference to earlier work called Iceroth lab that showed that dissociative agents tend to affect primarily RSC activity. We don't see this with the antipsychotics.
Thank you. And there's a question for Jerry, where the recent advances in using tools like MERFISH to potentially look at local protein synthesis, whether you've thought of potentially looking separately in axonal or dendritic compartments, whether you have resolution to do that.
It's nothing that we've looked at. Presumably, this might be something that your team is going to be interested in terms of the local expression levels on the axons and dendrites. I think these are things that are important. I think one of the things that I have to say about the work in terms of the cell type work and the molecular profiling is that right now, currently, all of these genes are just used as labels, right? We're just using them to kind of demarcate the different sets of neurons, but we have really little idea in terms of how these genes actually relate to the function of the neurons. And presumably, being able to understand these things, I think, will also help us a lot in terms of understanding what the circuits are doing, and that presumably will be guided if we can go into subcellular compartments, at the synaptic level, or at the axon and dendritic level.
So yeah, I think that would be super interesting in looking at the kind of categories of genes that are kind of unit specific, whether they are typified by various receptor channel, synaptic molecules.
Can I add a comment? On that?
Yeah.
So as Jared said, it's very difficult to use spatial transcriptomics like MERFISH method to identify transcripts that are in dendrites or axons, because it's just so spatial resolution is not enough. But people are now talking about potentially tagging or barcoding or tagging the transcripts that are transported into dendrites and axons. So maybe if there are ways performing cell type specific tagging of those transported transcripts, then we can sequence and identify them. Maybe you guys can try that. Yeah.
I wonder whether you could actually combine something like the expansion microscopy. With doing this type of more advanced MERFISH. That could be a way technically to get around that.
That's true.
So Honghui, there's another question for you from Matthew Larkham, where he says, at first glance, the cortex is distinctive because of the regularities, which same basic architecture, which has led many to intuit the understanding of a single cortical column would give enormous insight into the operation of the whole cortex. But your data suggests that there's much greater nuance to circuitry throughout the cortex. Do you think this means generalizations are doomed, or do you still hold out hope for a relatively simple abstraction?
I still think that's a great question. I still think that there is a general function that the cortical columns play. The variations that I describe are really just variations of the major theme. The laminar organization of the cortex is unique, compared to all the other structures, subcortical structures. Why is there a laminar organization in the cortex, and why is the cortex expanded so much in evolution and in its function, right? So we still don't know what a cortical column computes. Some great experiments here shown by Georg and Jerry, but we still don't know what a cortical column does. And I would love that people can find out. I think the variations I described are really just the simplicity or complexity of the layers, multiple layers. Some regions are simpler with fewer layers, whereas other regions have more subtle layers, and the variation of the cortical function is built into the layer, complexity of different regions and functional specializations. And there are specific cell types in specific layers of specific regions. But I consider that as all more detailed, fine-grained variations on the major cortical lamina organization. And I would love to know what it does.
So I have another question for Jerry, but I also want to say that you guys the panelists can ask questions of each other too. So, Jerry, I guess one of the other things that really characterizes this BAS1 phenotype is the specificity of the inhibitory connectivity. And I wonder whether some of the heterogeneity in inhibitory innervation could be kind of diagnostic of the cell types also. I guess I'm thinking about it because like in the work that we did with Idan, when we looked at, for example, thalamic inputs, we saw that different cells that were essentially the same cell type had really variable numbers and distribution of inhibitory inputs. And I think that if, different cell types have different connectivity that is, represented in the inhibition too, whether that could also be a diagnostic for their role in the circuit.
Yeah, so I guess what you're suggesting is maybe a kind of work from the connectivity backwards, maybe. So if you were to kind of extract certain connectivity patterns, which I think, on gross levels, I think there are differences. I mean, right, I think just the comparison between parvalbumin, that's, somatic, perisomatic targeting versus, somatostatin as dendritic targeting, right? But then presumably there might be even further subdivisions, right, depending on a subclass of SST or subclass of PV, and then you kind of work backwards from there to ask, well, is the excitatory innervation, how does that segregate? Presumably there might be, other patterns maybe that you might be able to define, a relationship you might be able to find, and if you go along that route, then maybe the route in which we kind of go in molecular to functional and anatomical.
Yeah, although I guess there could be more than one way to skin a cat. Yeah, okay. I guess Michael Lin also had a question for Honghui, but any can answer. How might the different pyramidal types develop their different connectivities? For example, are you seeing differences in synaptic adhesion molecules?
Yeah, so that's another important question. That's why we now study the development of cell types and see what specific genes are expressed at the time when the connections are formed. And we would be, for example, once we have the transcriptomic data, we would look for connected pairs and look for candidate adhesion molecules or something that are pairs of adhesion molecules, ligand receptor pairs that may be expressed in those connected pairs and begin to investigate. So I think there are good examples of the synaptic connection specificity, but I think a systematic investigation can start once we have comprehensive data sets. Others may have, specific ideas as well here.
I guess a question for Honghui related to that is the idea of experience and how that's going to play in everything. And how are you guys either controlling for that or taking that into account when you're looking into the developing cortex?
Yeah, so I think we are not doing experience manipulation type experiments right now. So we would love if other people can do it, because we usually just generate foundational data sets. We sometimes do some experiments, but we usually not. So we would love other people can look into that. Just wanted to say that, you know, in the developmental mouse visual cortex development data that we have collected, we see a dramatic change of gene expression profiles around eye opening. So before and after eye opening, there's a huge change in gene expression profile. So it could be very much related, and especially in layer 2-3. So it could be very much related to, Jerry, what you're talking about. The BAS1A neurons, some kind of things happened when the visual input comes in. The cortical circuits are reorganizing, especially layer 2-3 cortical circuits. So that would be, yeah.
So I guess there was another question, both for Jerry and Jorg, whether you've tested your predictions using optogenetics, or you could test your predictions using optogenetics. I don't know which of you wants to answer first. Yeah, go ahead, Jorg.
Sorry. The optogenetics, I'm not sure, I mean, not with the cell types that Jerry briefly talked about. So the RRAD PAS1A versus ADAMTS2, where we think they're positive and negative prediction errors, neurons, but we have made virus constructs to at least validate them functionally. So we know that they do have the, I mean, with all the caveats, and obviously, right, making viruses for a targeted gene, as everybody probably knows, isn't really straightforward, and it's not entirely clear how that 2 kb regulatory region inside an AAV actually relates to endogenous regulation. But I mean, short of this, all these somewhat murky problems that we have in validating this did work for these two cell types. They have the effects that we predict based on the molecular characterization. Optogenetically, it's not entirely clear, right? If the theory is correct, then these are positive and negative prediction errors. And it's not entirely clear what the consequence of shutting them down or activating them would be unless you know exactly what they code for, right? Even in a case where you just bulk stimulate, where you don't believe in predictive processing and think all excitatory neurons have the same coding capacity, it's not entirely clear what you predict to happen if you just, you know, light or excite all of layer 2-3. What the percept would be that generates, especially if you do it in visual cortex. So I'm not entirely sure how we would test that. I mean, we've been thinking about this, right? I think it's an excellent approach. It's just I have no clue how to properly do it and then show that, you know, that this is a functionally specific positive prediction error or negative prediction error neuron. So that the optogenetic testing, I'm not entirely sure how to do.
Yeah, it's pretty complicated. Jerry, did you want to say anything?
Yeah, I think, well, you know, in our current experiments, you know, we were only to kind of follow up on the cells post hoc. We didn't really have a genetic handle on it to be able to do some of these cell type specific manipulations. But with Georg's constructs, potentially, then that could be one way of kind of gaining access. The other way, maybe as Georg says, is like it might not be revealing to kind of just, you know, blast all the cell types at once, but you have to do something more subtle, right? If you can, I mean, we have ideas maybe if you kind of go more or less neuron by neuron and do some sort of manipulation and see what the consequences are on a circuit in a very, you know, task or feature specific manner. Maybe that's, you know, that might be there, that level of precision might be what's required than, you know, kind of more uniformly going after all the cell types are at once.
Mm-hmm. Rafa, did you want to say something?
Yeah, I think, I mean, I find both piece of work really inspiring and beautiful, no, and also agreeing with each other, providing maybe a path forward to understanding how the cortex works or what the cortical column is all about. But just to be fair, a critic would say, well, this is all correlation. There's no causation, and you still have to prove it. So this is a good hypothesis that needs to be proven. That's how I would put it. And optogenetics could be one way to do it. And I completely agree. It's complicated since there's so many different cell types. But for instance, the layer 5 idea that Georg was proposing, which was absolutely fascinating. No, I just couldn't wait to try to see what happens. Can you revert these symptoms by increasing or decreasing the activity of IT cells in layer 5? I mean, this is essentially what you're doing. It's funny that you're doing this with antipsychotics, which are notoriously dirty. I mean, this is you couldn't have picked a less clean pharmacology. So optogenetics, I think, gives you precisely the cell type entry, and that's pretty clean. Well, it's not perfect, but it's definitely a one way through. And I would just be fascinated to see what happens to these mice if you souped up or down the activity of layer 5 of these IT cells specifically. No? Yep.
Yes, absolutely. I mean, this is the type of experiment we want to do. I mean, I didn't show you, but I mean, it's precisely in the murkiness or the dirtiness of the antipsychotics that lies, I think, the power, right? Because we've in the meanwhile tested antipsychotic candidate drugs that don't even bind to D2. Everything, I mean, all FDA approved antipsychotics are somewhat related to D2 as partial agonists or antagonists, which is the primary hypothesis, hence the D2 hypothesis of schizophrenia. Anyway, we've tested now drugs that have completely different binding profiles. They have the same effect, right? Somehow we've tapped into a way to do something to these layer 5 cells with compounds with a variety of different receptors. I mean, I fully agree. We have found some dirty way by chance. All of these antipsychotics were discovered more or less by chance to do something to this network. And now the question is, can we do it in a more targeted fashion, right? This is exactly what we want to try to do. Can we slightly enhance the activity of all layer 5 cells? Can we slightly decrease? And this would then provide some gene therapy approach that directly targets the layer 5 cells without having all of these side effects that patients suffer from, because all of these drugs that we currently have are so dirty and target things in the heart, in subcortically, etc. Yes, absolutely. I mean, this is where we want to go. We want to find a way to do this, and it opened up the door to totally crazy experiments. Right, 2 years ago, if you had asked me, would it make sense to sort of try to turn off all of layer 2, 3? If this JEPA idea is true, right, something we're actually trying at the moment is silencing all of layer 2, 3, and if the testable hypothesis is now layer 5 should still function normally, it should just lose its ability to learn plastically. Right. So in this model, layer 2, 3 is just a teacher on top of the layer 5 associative network. And layer 2, 3 might be doing predictive processing. Layer 5 does something we don't fully understand. But this now becomes a testable hypothesis. So for us, I think it's exciting times. Things open up, and sort of the message is, right, the cortical column might be the wrong way to think about it. This is the sort of, I mean, and again, just to highlight, what I mean by wrong is when we think about the cortical column, we think about cortical layers 1 through 6 and don't include striatum, even though that's a very, very important connection, right? In the same way, I mean, layer 2, 3 might be horizontally the relevant computation that by all means talks to the underlying layer 5. That's why they're probably close together, but that the dominant computation is lateral, not vertical.
Okay, I think we're pretty much at time now. So thank you to all the speakers for a really fantastic session. I'll clap for everyone since we don't have a live audience. And we have half an hour now to go grab some lunch, and I'll be back at 1:45. Thank you.
Thank you.
I'd like to welcome you for the second half of this really exciting symposium, where we're looking at neuronal integration from circuits to dendrites. And this leg of the symposium, we have an amazing lineup of speakers. I'd like to warmly welcome Matthew Larkham from Humboldt University, Charité Berlin. Matthew is not only a maestro in multi-site dendritic patch clamp recordings, in fact, he taught me how to patch dendrites, but also of classical violin. And his lab has made seminal discoveries in defining dendritic spike types in rodents and humans. Matthew, take it away.
Well, thanks so much. And thanks to all the organizers. I'm really happy to be here. Actually, I'd rather be in New York, but I really like the format nonetheless. I think this is a great idea. So this is a symposium about circuits and dendrites. Circuits to dendrites is the title. And I guess when I look at it, I see it the other way around. Maybe it's just a small detail, but I see it really going from dendrites to circuits. And I guess the reason is that I think that when we talk about dendrites, I think we're really talking about computation. It's the not just the place where the inputs come to the neuron, but it's how the inputs are transformed essentially to outputs, which is really the definition of computation. And I think when we look at circuits, we're looking at networks. And I think the first question you ask, I think, should be what is the computational power of the elements of the circuit before you put them all together and work out what the circuit does. So I guess that's the way around I see it. So my title is From Top to Bottom, the Tough Dendrites of Pyramidal Neurons and Layer 6B, and this will make sense about halfway through the talk. So here you can see a diagram of the cortex, and I think Cahal knew, and all the early researchers just took it for granted. I think that all of these different neuronal types were different for some reason. But I think in the meantime, it's the recording site that's most common, almost ubiquitous across neuroscience, is the cell body. And I think that really skews the perspective of neuroscience to this day. And so you see a lot of circuit diagrams that look more or less like this. I'm just taking one of really thousands that I could have chosen. And as you can see, that when a cell type is being described in such a circuit, it's described as what looks like a point with arrows going from point to point. But of course, the reality is something more like this. This is the kind of schematic circuit I think that one has to have to really describe what's going on when you have real neurons involved, particularly in the cortex, which is a layered structure and has neurons that span the layers with their dendrites. And of course, there are lots of examples of this treatment of circuits. I'm showing one here from Rodolfo Linus, and Georg Keller was just showing us one. It's not that they're not to be found in the literature, but I think generally speaking, it's not usual to do this just because it seems too complex. And I think as great progress gets made in artificial intelligence, there's a lot of people wondering whether or not one needs to describe dendrites at all. But I think on the other hand, it's I think fair to say that to describe the cortical circuit, we're definitely going to have to include dendrites. And there's, I think, lots of converging evidence now to say that. We've been focusing on the tough dendrites, and I wanted to focus somewhat in this talk on why that seems an important place. It's a kind of unlikely structure because it's so far away from the soma, and you wonder why there should be manifest input to this. But the data that Mike Hausser was just showing attests to its importance. Every time I see Mike's data, it just blows me away. It's even more incredible every time I see it. And it's I think always been just a remarkable fact that these neurons are really not the way I think an engineer would make it, and it begs to be explained. We had several, I think, convergent findings recently. One was that if you anesthetize an animal, the tops of the neurons, the tough dendrites, get decoupled from the cell body, and that might well be the explanation for anesthesia. And there seems to be some sort of nexus point that's dictating whether or not feedback that Mike was so beautifully showing that tends to come to the top compartment can be regulated on its way to the cell body via the input from the thalamus, some sort of nexus point there. And that might constitute some sort of third compartment. And another study that we were recently working on suggests that the layer 1 and the tough dendrites of pyramidal neurons is the site of associative memory or semantic memory. I won't go into the details here, but I think it makes a lot of sense if the columns are coding for features and feedback is coming to the top of the columns, then it would make a lot of sense that the associated activity around the brain that needs to be learnt, that corresponds or is coincident with any given feature, should be in the top of the cortex. And so we're currently pushing this as a hypothesis, and there was some great work done in my lab by great postdoc Guy Doron and a PhD student, Jun Shin, who's presently in New York, and they were showing that that you could stop learning by blocking input to layer 1. Anyway, not hanging around on that. This kind of generalizes to this kind of a view of the pyramidal neuron in the cortical column. I'm talking now about the layer 5 pyramidal neuron, the output neuron of the column. And I think we're at least converging on a let's say an abstraction of this cell having 3 compartments. One, the apical compartment is able to have various kinds of nonlinear events, and the basal compartment receiving feed-forward feature-specific information, and then some middle compartment that's dictating whether or not these 2 things are coupled. And in the focus on the tuft, as Mike was alluding to, and several people have alluded to, we think that there are so-called NMDA spikes there. And the important thing about NMDA spikes is that they rely on so-called clustered input. So it relies on there being a convergence of synaptic input onto some branch, such that you get a regenerative event that's orders of magnitude more than the combined input itself. So it generates active conductances in the dendrite that are like mini explosions in the dendrite. And the idea would be that there's convergences from these horizontally running long-range fibers that are coming from all over the cortex and from all over the brain, and that essentially this is a detection device for what else is going on, such that it can be associated with the feature that it needs to be associated with. So we looked at this in vivo, like Mike has done, and Dr. Schiller has done, and several people now in the tough dendrites, and could show that there are local events. And I think there's a convergence now on the distance of approximately 10 microns, up to 20, maybe 30 microns in the widest cases, but very local. And local means that there's a really complex computation going on in the dendrites, which is very different to the other kinds of active events, like the action potential that's generated in the axon, or the action potential that we know is the calcium action potential that can be generated in the apical dendrite. And these events are very well suited to capturing, let's say, coincidences of very few synapses, and that means very few other firing neurons around the brain, such that it could be, let's say, some sort of coordinated event. So I apologize for this horrendous list here, but the point about this is that we now have, I think, a trajectory of just over 2 decades looking into this particular neuron and what it's doing. And we're converging, I think, on, first of all, on the notion that it's coding the output of the column, which anyway, I think is the anatomy speaks to. And we would conjecture that what the information that it gives to the rest of the brain is the probability of the feature that column is calculating, given the context, and the context is coming in through the top of the neuron. So in some sense, the feature is grounding the cortex and these neurons and the cortical column to the outside world, whereas the information coming to the top of the neuron is kind of like the content of either your memories or the current, if I could put it this way, thought that's going on. And then added to that, there's this middle compartment that is somehow regulating how attached you are, if you like, to the top of the cortex. And this could be something to do with awareness, as in why you lose it with anesthesia, but also attention, as into which parts of the brain you're currently paying attention. Now, so all of that is just the warm up or the background to a new study that might seem at first glance to be shockingly off topic. So I've got a really talented postdoc at the moment, whose name is Tim Zolnick. He's been with me for a while, and he's been working on the lowest layer of the cortex, which is, I think, officially called 6B at the moment, although some people have called it layer 7. Cahal called it layer 7, for instance, and it oscillates in and out of being called layer 6B and layer 7. It's a very thin layer. It's only about 5% of the cortical column neurons, and right at the bottom of the cortex, it's been quite ignored. There's not many studies on this, although it's been known for some time that these are the orexin sensitive neurons. That's to say, they're arousal neurons, if you like, and they're the only neurons in the cortex that are sensitive to orexin. And they're also sensitive to a whole heap of neuromodulators. And if something goes wrong in the orexin sensitivity in your brain, you get narcolepsy, and you just impulsively fall asleep the whole time. So they're somehow really important for maintaining wakefulness. So the first thing that Tim did was to look at what projects to layer 6B. And here was the first surprise for us, which was that although thalamus well and truly inserts into basically all layers of the cortex, it doesn't touch layer 6B, or there's very, very minimal input from the thalamus to this bottom layer. It's the only layer that gets virtually nothing from the thalamus. And on the other hand, it gets a whole heap of cortical inputs and quite long-ranging cortical inputs. Actually, the reason I really wanted to work on this layer, and so I was encouraging Tim for some time to go on with this study, was that we wanted to look at the output of this layer to see what it really did. And we've been known for some time that this layer projects to layer 1, which was, for me, always a smoking gun. Anything that projects to layer 1 must have some sort of significance, I think. And here you can see a TD tomato expression in layer 6B neurons. There are now, I should say, two transgenic mice, which make it much easier to study this layer. So this is one of them, so-called DRD1 Cree mouse, and so you can see if you express TD tomato in those that there's, first of all, there's a very, very dense labeling of layer 1, and secondly of layer 5A. There's also a projection to higher order thalamus, and virtually nothing in the first order thalamus or the thalamic reticular nucleus. So it avoids that altogether. And so that's quite a remarkable output. The thing that's so remarkable about that is that it's completely and utterly overlapping with the projection from the higher order thalamus. So here you can see it's not just going to layer 1 and layer 5, but to layer 1A and layer 5A, and which is also true of higher order thalamus, completely overlapping. So in a sense, you've got a second thalamus in the cortex that is projecting to the same layers. But we really want to know not just which layers it projects to, but which neurons it projects to and what it actually does. And so Tim's been working away very hard in the past few years try to get a hold onto this. And one of the things he's done in the meantime is to come up with I think a really ingenious method. It's kind of based on the circuit mapping techniques that are existing at the moment, which are use channel rhodopsin. And so, for instance, the main one is Crackham, which was developed in the lab of Karel Svoboda. And that's Crackham for channel rhodopsin assisted circuit mapping. And this is the same kind of principle, but instead of using an electrode on the postsynaptic neuron, Tim had the idea to use this optogenetic construct called Campari. And this is a construct that essentially is an activity measurement protein, in that it starts off green when there's no activity, and it converts to red when a cell has activity. In fact, it's measuring increases in calcium in the postsynaptic cell. So using the same, so by taking, let's say, a Cree transgenic line, you can put channel rhodopsin into the projection neuron of your choice, and then see what neurons get activated on bulk, and basically save yourself a decade of paired recordings by looking at which neurons went red all over the brain, and in particular in the column. And so here the Cree starter line, if you like, is the layer 6B neurons. So they're the ones that are being excited. Their axons are being excited in this approach. And here you can see, so these are all the neurons expressing Campari in the beginning of the cortex before there's been any light. After light, we can now look at what neurons have converted to red. And you can see immediately that there's almost nothing being excited in the upper cortical layers, but there's quite a lot of layer 5 neurons. And then just a few spurious, what turns out to be interneurons. And the other nice thing about this method is in post hoc, you can use immunocytochemistry to find out what these neurons were. So we can basically see that it's almost exclusively targeting pyramidal neurons, layer 5A and layer 5B pyramidal neurons, which turn out to be both IT and ET, or this are the intratelencephalic and the extratelencephalic pyramidal neurons, and then a few deep layer interneurons. And that's more or less it for the output of this layer. So summarizing that, you've got input from just cortex to cortex, as it were. It only receives cortical input, ignores the thalamus altogether, but it behaves like a thalamus, projecting to the pyramidal neurons, specifically in layer 5A, predominantly, both at the top of so in layer 1 on their tufts, and in this really crucial middle compartment that we think might be connecting the two neurons. So if you stand back from that, one way of interpreting what's going on is that the layer 6B is impinging on the thalamocortical loop, and we and others, I think, would say that the key fact about the thalamocortical loop is it needs to be engaged for you to be aware, to be aware of, to have let's say, a conscious thought, or for something to pop into your awareness. And this layer 6B is not just hitting the major nodes in general terms, but in really specific terms. So it's going to both of the nodes of the neurons that are projecting to higher order thalamus and coming back. So the complex way of describing this is in this circuit here, which is more information than you need to know right now. But basically, it turns out that there are 3 populations, and then that they subdivide in terms of which ones go to which compartment. So there's a whole lot of control that 6B has. We don't understand yet what projection neurons are causing which populations here to do their thing. There's a third population, by the way, that goes to the superior colliculus, which is also, when you look at it, when you stand back for it, really part of the thalamocortical loop. So now getting down to brass tacks, and trying to find out what this actually does to the neurons, this is quite astonishing. So first of all, the 6B input to the tough dendrites has a massive effect. So this is, we've got channel rhodopsin in their axons, and we're putting the beam of excitation light onto the axons around the tuft of these neurons. And basically every pulse, this is many pulses, about 10 pulses in a row, or most of the pulses cause huge events that are clearly NMDA sensitive. So this is what you get if you block the NMDA channels. And there's something left, which is presumably the AMPA, and that tells you, I think, what you would get at the cell body if you just had passive conduction and passive generation of EPSPs in the tuft, which is showing you just the massive difference. And it's so big that it can cause the cell to fire on single pulses. Now we can compare that to what the thalamus does, because remember that's projecting to exactly the same place. It also has a large effect, but not quite as large as 6B, and that's also very NMDA sensitive. So that tells us, I think, that all of this control is going through active events in the tuft. I was talking there about the extratelencephalic neurons, and Tim also did all this work on the intratelencephalic neurons, where there's also large input that's also NMDA sensitive, not quite as large as to the ET neurons, but still very large. And there's an argument there that maybe the thalamic input to the IT neurons is slightly larger than the thalamic input to the ET neurons. So putting all that together, basically, we think that you've got a second thalamus that's basically inside your cortex that probably you're not aware of. We're speculating that this is one that's sort of subliminal, and maybe, as Tim likes to suggest, is sort of the gist idea that, you know, when something's on the tip of your tongue and you don't quite know what it is, that this is that you're somehow generating these signals with a cortical input, and it's not until you engage the thalamocortical loop, which this does rather nicely, that you suddenly have that thought in your conscious perception. And you've got to imagine, so this yellow ring here is the 6B that's at the bottom of the cortex that's surrounding, if you like, the hub, which is the thalamus that's basically the conductor, and that's presumably sending signals all around the brain. And you've got another one inside your cortex, as far as we can see, and you can only describe the effectiveness of this if you make reference to the dendrites of the pyramidal neurons. So lastly, Tim did one sort of proof of principle experiment.
5 minutes,
Okay,
Matthew.
I'm right at the end, actually. The one proof of principle experiment to look into this, and we had the idea that what you could do, since we know that thalamocortical loops are engaged in states of consciousness. One of these is the sleep state, and of course layer 6B is known to be very tightly regulated by sleep modulation. So he basically sleep deprived mice and nevertheless put channel rhodopsin in the axons of layer 6B neurons, and he can see a head fixed mouse with a light guide coming down onto the craniotomy, and so the light is being shined onto the axons of the 6B. Every time there's a blue dot, the light was shining, and you can see subsequent to that there's whisking. If you look at the electrical recordings, it's just astonishing. So during the normal periods, because it's sleep deprived, you've got really high so-called delta activity, so slow wave sleep. And just for the time of the light, you go into high gamma, and then as soon as the light's gone, you come out of high gamma back into basically this sleep kind of activity. And you can see here the light really increases high gamma, and it really kills the lower frequencies. So summarizing that. Basically, the idea is that this is some sort of wake-up signal for the brain, and it's going directly through the layer 5 neurons. Again, reiterating, I don't think you can understand this without reference to dendrites and dendritic properties. It would make no sense to do a wiring diagram to just some cell bodies in layer 1. This has to be doing something really incredible at the level of the dendrites and information processing. And well, that's one way of summarizing. The other way of summarizing this whole talk is that basically the top and the bottom of the brain are somehow really sandwiching all the other all the other cortical layers, and are both, I think, enigmatically important for understanding the cortex. So that leaves me to thank very much Tim Zolnick for all the work that he's put in over now quite a few years, and it's really coming to fruition at this point, and a whole host of other people that have been involved in this story over many years, and you for your attention.
Thank you, Matthew. We'll take questions during the panel discussion. So we'll move forward to Yota now. I'd like to warmly welcome Yota Poirazi. She's been a pioneer in dendritic computation. She's been building models at many levels of abstractions, from very detailed biophysical models incorporating circuits and cell types to also introducing, bringing the idea of dendrites to the artificial neural network landscape with this whole idea of multi-layered ANNs. I'd like to warmly welcome Yota, who's been a role model and a leader in the field, amalgamating all the dendrites people.
Thank you very much, Zakita. It's great to see everyone. Thanks for the invitation. I hope you're looking at my slides just fine. Already Michael and Matthew did a great introduction about the importance of dendrites, so I won't say much about them and get right into the point. But I would like to start with this slide because we're talking about dendrites and circuits, and this is what happens to our brain across age with respect to the dendritic morphology. And as you can see here, we start out having very simple dendrites. We have complicated dendrites around the age of 30 to 60, which is also the peak of our cognitive abilities. And then as these abilities start to decline, so does the dendritic morphology. So that's an important reason why we should understand the dendritic computations and the dendritic morphology, let's say. Now, there are many different things that dendrites do for the way our brains compute. I don't have much time to go into them. I will briefly mention a few and then just focus on my favorite subject. For example, as Jackie Chiller and many others already shown many years ago, dendrites allow individual neurons to amplify their signals through the generation of dendritic spikes. And many times these dendritic spikes are only generated when the signals arrive in very short time differences, so nearly coincident. And these are two types of nonlinear computations that are afforded by dendrites. This kind of dendritic spikes, as Matthew already explained, are often affected by the way inputs are activated. So the organization of synaptic inputs into nearby locations and the formation of synaptic clusters is a very efficient way for driving strong nonlinearities like NMDA spikes. Because of this organization of inputs into clusters that are often found within individual dendrites, we and others have often suggested that individual neurons can act as nonlinear integrators, where each one of the dendrites has its own nonlinearity, underlined by the generation of dendritic spikes. And this is important because we can then think of neurons as small neural networks. If you think of every dendrite as an individual nonlinear unit, because of its thresholding abilities, then you can think of a neuron as a 2 or 3 or multi-layered neural network. And many studies, both theoretical and experimental, are building on this notion. So these are just a few of the contributions that are being discussed in the literature for many decades for dendrites. Now, I want to focus today on synaptic clustering and synaptic turnover. And synaptic turnover is essentially the changes in the formation and elimination of spines within dendrites. And I want to discuss their possible contributions to learning and memory functions in biological and artificial networks. So by synaptic turnover, as I already mentioned, this is a phenomenon that happens routinely at baseline conditions where spines are grown and deleted. They are stabilized depending on their ability to make postsynaptic partners that are important in the sense that they carry important information, so they're frequently active. And if something like this doesn't happen, so a postsynaptic partner is not active, then these spines can withdraw and be deleted. And this is, an ability or a phenomenon that occurs throughout life. It's mostly pronounced during development, but it's also seen during adulthood. So we showed many years ago when I was doing my PhD with Bartlett Mel that if you build a computational model that has this ability to grow and withdraw synapses, and these synapses are found within active dendrites, then if you train this network to store patterns, these inputs will move around and you will end up having synaptic clusters within dendrites. So turnover is one of the mechanisms that can drive synaptic clustering. And this is important because if you have this phenomena, then you build models that are capable of storing much more information than if you have just linear dendrites and no synaptic turnover. So this is a very old theoretical study. I'm only mentioning it here because many years later, we started looking into these theoretical predictions. And I want to take you now to a study that was done by the lab of Alcino Silva back in 2016, where they looked into the mechanisms that underlie the linking of information across time. This may sound completely irrelevant to synaptic clustering and turnover, but I will get there in a moment. So what Denise did, which was a brilliant postdoc in the lab at the time, she exposed animals into different contexts. Here, the green, the red, and the blue box. And she separated these contexts by different time intervals, 7 days versus 5 hours, for example. And she delivered a fearful stimulus in the blue context, the one that was separated by only 5 hours from the red context. The hypothesis was that if you have experiences that are close in time, let's say separated by only 5 hours, then there should be some cellular correlate of this association between these two experiences that should be linked across time. So after the behavioral experiments, they looked at the population of neurons that encoded the different contexts, and they looked for population overlaps. And what they found was that there were significantly more common neurons, so higher overlap in the populations that encoded the red and the blue context, the two contexts that were close in time, compared to the contexts that were distant, separated by 7 days. And at the same time, these two memories, the ones that were overlapping, they were also associated at the behavioral level. So the animals that were exposed to the red box exhibited freezing, so fearful behavior, because they were food shocked in the blue box, the one that was separated by 5 hours from the red box. So there was both a behavioral phenotype that showed that these two memories were linked, and also a cellular overlap that was proposed to be the mechanism that underlined this linking of the two memories. Now, we built on to this study together with Alcino using a computational model in order to try to understand how do these common neurons that encode both of the linked memories become overlapping? What could be the subcellular mechanism that underlies the overlapping neurons? So to do that, we built a computational model, which I will depict to you in a video in a second. So this computational model was furnished with several plasticity rules. I don't have the time to go through them, but you can find them in the study. This is already published. And we had nonlinear neurons with active dendrites, pyramidal neurons, and interneurons in the model. And we used it to simulate the exact same behavior that Alcino and his lab did in mice. And what we wanted to find out was what are the subcellular mechanisms that may explain this memory linking. So one of the properties that we included in the model was an increased excitability of pyramidal neurons, which is known to occur after learning. And this increased excitability lasts for several hours. And what we found out was that this increased excitability could explain why the same neurons, the ones that learned the first memory, were also highly likely to capture the second memory because their excitability was higher. And we replicated the observation in mice, where they found that about 20% of the neurons were overlapping when they experienced two memories separated by just 5 hours. But what we did with this model is to also look at what happens in the dendrites of these common neurons. And we found that there were also common dendrites that encoded the two memories. And within those dendrites, our model predicted that we should find spines from both of these memories that are located close to each other, located within the same dendrite. And we even predicted the size of these small clusters. They would range between 2 and 5 synapses. And this was what we thought the subcellular mechanisms, a mechanism that enables the two memories to become linked, first at the dendritic level, and because they are co-allocated within the same dendrites, when they're activated together, they drive activity in the same neurons, and that's how the memories become linked. And this was a prediction generated by our computational model, which you can also see here. In this simulation, essentially, we placed inputs from 2 different memories, the yellow and the blue, randomly allocated onto the dendrites of many neurons. And once we activate the first memory, which you can see here as the yellow spines going off, then they drive calcium spikes within the nonlinear dendrites of these model neurons. And this increased activity results in plasticity proteins to be produced and plasticity processes to kick in, and these inputs are strengthened, forming small clusters of the first memory. This increased excitability also drives a higher activity into this particular dendritic branch, which remains there for some time. And when the second memory comes in, which you can see in blue, now it's capitalizing on the increased excitability and the plasticity that is available in this branch and creates even more clustering through the strengthening of the blue synapses. And that's how our model predicts that we link these two memories across time. So then, recently, Megha Sigal, a very talented postdoc in the Silva lab, went on to test this prediction in experiments in mice again. So she pretty much repeated the same experiments that Denise did, exposing the animals into different contexts, again separated by 7 days versus 5 hours. But this time, through a very fancy molecular technique that I don't have the time to explain, she was able to tag the dendrites that are storing these particular memories. So instead of looking at population of neurons, she was looking now at population of dendrites that are active when these two memories are formed. And again, she found a much higher population overlap, in this case, the dendritic overlap, when the two memories were separated by 5 hours versus 7 days. Here you can see the overlap, which is between 15 and 20% at the 5 hours. And in our model, surprisingly, we predicted a similar, let's say, dendritic overlap around, sorry, this should be 15, around 17% at the dendritic level. So this was a very, let's say, nice confirmation that the model prediction was largely correct. So Megha also went on to look at the second prediction, which concerned the presence of clusters of spines within these common dendrites. So she kept track of new spines that were formed within a distance of less than 5 micrometers next to an existing spine. In the apical dendrites of pyramidal neurons, this is all done in the retrosplenial cortex. And she plotted these two observations for the two scenario of memories being separated by 7 days versus 5 hours. And what we see here is essentially the addition of new spines following exposure to the first context, context B versus context A, so the difference is 7 days here, or context B versus context C, the difference is 5 hours here. And what she expected to see, if our prediction was correct, was a stronger correlation in the 5 hours versus the 7 days experiment, which she kind of sees here. Of course, it's not a very strong correlation, but there is some indication that the newly formed spines are preferentially taking place within dendrites that encode the first context when these two contexts are separated by 5 hours, and this is not the case when they're separated by 7 days. So there is some support, again, for the modeling prediction with respect to memory linking and the occurrence of synaptic clusters. So the take-home message from these two studies was that both models and experiments suggest that synaptic clustering in active dendrites may be one of the mechanisms that underlies memory linking across time. So another reason why synaptic turnover is important was also shown by the same lab a few years back, when looking at mice that had this mutation in the CCR5 gene, and this particular mice had the ability to learn faster. So the CCR5 animals were trained in a fear conditioning tasks over days. And as you can see here, compared to wild type, they were able to freeze, so to learn the fear conditioning task faster than the wild type. And what was interesting about these mice was that because of this mutation in the CCR5 gene, they had higher synaptic turnover in their baseline condition compared to wild type. And they were wondering whether this higher synaptic turnover could be the reason why these animals learn faster. What they observed post-learning was that these animals, presumably because of the higher synaptic turnover, they also exhibited higher clustering of spines within the dendrites of pyramidal neurons. I think it was layer 5 in the retrosplenial cortex again. So they had more cluster spines, and these cluster spines had a better survival compared to the wild type animals and the non-clustered spines. So this experimental data essentially suggested that increased synaptic turnover could explain faster learning in these animals. And the reason why learning was faster could be because we had more cluster spines being formed as a result of learning in the apical dendrites of pyramidal neurons in the retrosplenial cortex. So we teamed up again to use a computational model to see whether we can explain these observations at the mechanistic level. So in the same computational model, we essentially added synaptic turnover and increased the rate of the synaptic turnover by increasing the number of dendrites that had this higher rate of synaptic turnover. And we observed whether in this computational model, the formation of a memory also resulted in higher clustering, which is what we found in our model. But we also looked at the properties of this memory engram. So how sparse was this memory if we increased the synaptic turnover? And by sparse, we meant that the neurons encoding the particular memory had an activity which was very different compared to the rest of the neurons in the population. So what we found was that as we increased the number of dendrites with synaptic turnover, we had sparser and sparser memories. Sparser memories mean that you have a higher ability to discriminate them from noise and a higher computational storage capacity. So essentially, our model predicted, compared to together with the experiments, that perhaps increased synaptic turnover leads to faster learning through the formation of more clustering and the ability to generate sparser and more easily distinguishable memory engrams. So the take-home message from these mixed computational and experimental studies was that synaptic clustering is an important feature, because by effectively driving the dendritic spikes, perhaps could be the mechanism that drives the linking of information across time. That synaptic clustering in active dendrites could be one of these mechanisms that underlies this linking. And by facilitating synaptic clustering through synaptic turnover, we may have improved learning and storage capacity. So the question that I want to focus on in the last part of the presentation is whether this synaptic turnover can also help machine learning systems, so artificial neural networks. So what we did in this work is to take the model that I presented just before, so the biologically inspired neural network, which is all essentially based on spiking neurons with active dendrites, and change it so that we can use it to learn a machine learning task, in this case, the classification of MNIST digits. Now, the change that we did in this network essentially was to group our neurons into two populations. We did this because we want to discriminate digits organized in two classes, so zeros from ones, for example. And we also gave a small teaching signal to this network, which I will explain in a second. Our neurons are composed of cell bodies and dendrites. The dendrites are nonlinear based on our experimental data from years ago. We have two types of interneurons projecting at the cell bodies and the dendrites of the neurons. And the way we use this model to learn to discriminate digits is essentially to take the image of the digit and translate the pixel, every pixel in the image, into a spike train. And the firing frequency of this spike train is essentially determined by the intensity of the pixel. The higher the intensity, the higher the frequency of the spike train. And this is delivered as an input to all the neurons in our network. But we have two dedicated populations. One will be trained to discriminate zeros. The other will be trained to discriminate ones. And this is done by pairing a teaching signal to the first population only when zeros are presented, and pairing a teaching signal to the second population only when ones are presented. So we're essentially using the properties of this network to form associative memories in the same process that we learn to bind information across time, now to discriminate inputs, to discriminate digits. Now, the learning rules in the model are the same ones that we had before. So we have a calcium-based LTP and LTD rule. We have synaptic turnover, which is the mechanism of interest in this particular study. We have a stopping criterion, which essentially decides to stop learning when the synapses reach a particular size, based on experimental data that tell us that spines grow up to a particular size and then can no longer grow further. And we also have an adaptive learning rate, which means that small spines grow faster than larger spines. These are all biological learning rules implemented into the model. And we want to explore how synaptic turnover affects the learning abilities of this particular network. So what you see here is the performance on MNIST classification. So discriminated 0 from 1, 0 from 2, and so on and so forth. This is a binary classification problem as a first step. And the upper level of the figure here illustrates the performance when we have synaptic turnover. The bottom half of the matrix illustrates the performance without synaptic turnover. And the lighter the pixels, so closer to yellow, the higher the performance. So you can already visually see that if we have synaptic turnover, so in the upper triangle, we have a significantly higher performance than if we don't have it in this particular network. And you can find more information in a bioRxiv paper that is listed here, the QR code here. So turnover improves performance, but the question is, does turnover improve performance at any given condition? And based also on the fact that the brain has limited amount of resources, so a limited number of synapses that can be plastically modified, we also looked at what happens when we increase the number of resources, so have more plastic synapses, or versus decreasing the number of resources, so cutting the number of synapses in half. This is the control condition in the middle, where we just show that we have better performance with synaptic turnover. If you look at the condition with double the synapses, then the performance advantage is not really evident. But if you look at the condition when you cut the synapses in half, then the performance advantage is significantly higher. So this suggests that synaptic turnover is an important mechanism, especially when the network has only a few synapses, so a small number of plastic synapses. In this case, the control situation had 7,500 synapses. Here we have 3,500. The double is 1,500. Please keep in mind that this is a small network. We only have about 100 neurons in this network, and that's why we're also focusing on a binary classification problem. So the other thing that we wanted to look at is whether synaptic turnover is important when the task is easy or difficult. So what we did here is to group the digits into difficult task. For example, separating 4 from 9 is a difficult task. An easy task is separating 0 from 1. There are some pairs that are easier than others. And we looked again at the 3 conditions. So double the number of synapses versus half the number of synapses with and without synaptic turnover. Again, if you have a lot of synapses, it doesn't really make a difference whether you have synaptic turnover or not. But if you don't, if you have a few synapses, then it makes a huge difference if you have synaptic turnover. And it is consistently better for all levels of task difficulty. So it doesn't seem to make a great difference if you have a very easy versus a very hard task. The other thing that we wanted to look at is how fast is learning. We've seen already from the experiments in the CCR5 mice that animals with high synaptic turnover learn faster. So we wanted to see whether in an artificial system, this is also the case. And what you see here across all 3 conditions, irrespectively of whether we have a few or a lot of synapses, with synaptic turnover, you have a much faster learning. So blue is with synaptic turnover, and it is easy, it is consistently a much smaller number of epochs that is required to learn the task compared to the no synaptic turnover. And in this case, fewer epochs also means fewer images, because every epoch in this network, it is a presentation of an image. So not only the network learns much faster, but it also requires a much smaller input data set to achieve a very similar performance, the same performance, in fact, in this case, with synaptic turnover. So this is a very important advantage that is provided to the network when we have synaptic turnover. Now, is this advantage important in the presence of active dendrites, or do linear dendrites suffice? What we did here is to use linear versus nonlinear dendrites in the network and just compare the performance without modifying any other parameter. So without extensively tuning the linear network. And you can already see that without having nonlinear dendrites, the benefit is significantly reduced. It is still much better with synaptic turnover versus without having synaptic turnover, but having nonlinear dendrites definitely boosts the performance in this network. And the last thing that we wanted to understand is where do these benefits come from? So we went in and did a more extensive analysis to see where these benefits come from. I think I will skip this slide and go straight to the representations that are produced by the two networks. So what you see here at the top is the images that the networks produce after training. Once you look only at the resulting weights, at the no turnover case versus the turnover case, and as you can already see from these two representations, without having synaptic turnover, the resulting images are highly noisy. So the network is never capable of reaching a very high resolution representation of the input digits, whereas when it has synaptic turnover, this representation is much more clean. Now, why is this representation clean? We went in and looked at the distance of the resulting representations from the class. So the class of zeros versus the class of one. And if these representations are very high quality, then the distance from the class should be as small as possible. A small distance means a high similarity score. So what you see here with synaptic turnover in blue is that the similarity score is consistently higher with synaptic turnover versus the cases where we don't have synaptic turnover, even for the tasks that are very difficult, and therefore the representations are not as high quality. So this analysis already tells us that having synaptic turnover enables the network to produce higher quality representations. The other important feature why synaptic turnover may work is generated by looking at the synaptic weights. So we have a lot of synaptic weights in the network, but not all of them get used. If we don't have synaptic turnover, which is the blue case here, a lot of synaptic weights remain unused. They are not effective. The turnover is the blue one. So the yellow is without synaptic turnover, and a lot of the synaptic weights in this case, they remain ineffective. Remain ineffective means that they don't have a very high value. So you see that if you have synaptic turnover, many of the weights become plastic, and this is probably because by moving these weights around, you allow the network to find those presynaptic axons which carry important information for the task at hand, in this case, information related to the specific digit. If you don't move these weights around and they remain fixed, it may happen that from the beginning, these axons do not have important information with respect to classification, and therefore these weights are not updated and they remain small. So this analysis suggests that if you have synaptic turnover, you have better representation of each class, irrespectively of the difficulty level. And this is probably because you have better utilization of synaptic weights. This is also seen by looking at the distribution of weights with turnover versus without turnover. We start with random weights, and therefore a lot of them have small values. After learning, these are the distribution of the synaptic weights, and you can see that a lot of them remain small. They are not utilized if you don't have synaptic turnover. But with synaptic turnover, all of the weights in this distribution are relatively uniformly distributed, which means that they are more effectively utilized by the network. So in summary, what I presented is a spiky neural network model, which was used both for experiments to explain how we can link information across time, for example, to explain how synaptic turnover can increase the sparsity of a learned representation and therefore facilitate storage capacity. And this same network model was used to solve a machine learning problem, in this case, MNIST. Importantly, the network uses purely biological learning rules. So there is no backpropagation, there is no classical error propagation in this network, and it's still capable of learning a machine learning task. We've shown that synaptic turnover improves learning, both in terms of performance accuracy, but also with respect to the robustness of learning, so having better representation of the different digits, and importantly, the speed of convergence. And we've shown that synaptic turnover is especially beneficial under conditions of low synaptic resources, which could be a reason why this mechanism has evolved in the brain, where the size of the scalp and the resources are limited. And finally, we've shown that synaptic turnover improves learning in this machine learning task by facilitating the optimal usage of trainable weights, resulting in better class-specific representation and better utilization of these weights. And with that, I'd like to thank the people of my lab that made this work possible, especially George Kastelakis, who was the guy who developed a brilliant postdoc. He developed the original spiky neural network that was used in the studies in collaboration with the Silva lab, and Nikos Malagassis, who was a master student in the lab and is now doing his PhD in Germany in the lab of Juliana Georgieva. He did the machine learning work with the help of Spiros Halvlis, a very talented postdoc in the lab. So thank you very much for your attention, and I'm happy to take any questions.
Thank you, Yota. This was an incredible lineup of talks. We actually talked about nonlinear computations in the dendrites and how feedback inputs can drive branch-specific activity and dendritic events, how layer 6 arbitrates arousal and oscillatory state transitions, and thinking deeply about how apical dendritic activity could actually allow for that to happen. And then Yota just spoke to us about the turnover of spines and how it boosts sparsity of learned representations and improves performance. We have a few questions already lined up. Let's start with those, and then I hope we can get into a very lively discussion following these questions. So there is, the first question is from Felipe Godoy Guejardo, and it's for Mike Hausser. First of all, thanks for your presentation. Considering that this dendritic activation, it's branch specific and compartmentalized, this phenomena depends on differential expression of calcium permeable channels, or it's an intracellular calcium reservoir event. What could be the role of intracellular calcium reservoirs? What's the role of spine apparatus protrusions in the ER that's present in the soma?
Okay, first of all, great question. And I could talk about this question for hours, but I'll try and keep it short. So we know a lot about the biophysical basis of these local dendritic events from patch clamp and imaging experiments in brain slices. So this is the work of Jackie Schiller, Greg Stewart, Jeff McGee, Matthew Larkham, Nelson Spruceton, and many others. So I highly recommend you go look at their papers. What we know is that these events require a combination of voltage-gated calcium channels in the membrane and NMDA receptor activation. And different combinations of these two can then give you either dendritic calcium spikes or dendritic NMDA spikes, or some mixture of the two. Okay, and so these two sources, these two channels, trigger electrical events that cause the initial calcium signal, the calcium entering into the dendrite. But there could be additional contributions from, for example, calcium release from intracellular stores from the spine apparatus, as you suggest. So that's certainly something that could boost the calcium signal that's coming from the calcium channels or from the NMDA receptor activation. And by the way, one important aspect that you highlighted, modeling studies show that the calcium channels, the voltage-gated calcium channels, don't need to be non-uniformly distributed to produce these local events. You don't need hotspots of calcium channels to get these local events. You can get it even with completely uniform channel distributions. And by the way, this distribution of the channels is one of the big unknowns of dendritic physiology. So the quantitative subcellular distribution of proteins across the dendritic tree is basically unmeasured. It's incredibly hard to measure, especially in a high throughput way. So we know very little about the distribution of channels and receptors on a quantitative level. So hopefully, the combined program, maybe via the collaboration of the symposium organizers, can eventually provide us with this important information. Thanks for the question.
Thank you, Mike. There's one more question for you. It's from Nikolai Dembro, and they ask that, do you predict that pairing the spines responsive to the LM feedback stimulation prior to or after a global event would shape their propensity to recruit subsequent local or global events?
Hi, Nikolai. Yeah, that's also a great question. So we didn't actually do this timing experiment yet, but it's a very cool suggestion. And I think you might expect effects on two time scales, on short and long time scales. So on the very short millisecond time scale, Matthew Larkham has shown that layer 5 pyramidal cells exhibit a form of coincidence detection called backfiring, right? Where the apical tuft inputs can non-linearly interact with backpropagating action potentials over a roughly 20 millisecond coincidence window to enhance the impact of dendritic inputs on the soma. And so this can trigger an additional backpropagating spike, resulting in bursts of spikes, and ultimately a kind of dendritic somatic ping pong that can kind of reverberate within the cell. So that's on the short time scale. On the longer time scale, the local calcium signal can drive synaptic plasticity, which in turn can enhance the future probability of triggering these local events. And Katie Bittner and Jeff McGee have shown in very beautiful work in hippocampus that the temporal requirements for such plasticity can actually be remarkably broad over several seconds. So if similar mechanisms operate in cortex, this leaves a big time window for this kind of pairing leading to synaptic plasticity.
Thank you. There's a question from Matthew from Fritjof. How columnar or horizontally widespread is layer 6B output?
Quite wide. It's so stimulating in somatosensory cortex. You can, so let's say if you've got the sleep-deprived mice and you do the stimulation in somatosensory cortex, as I showed, but do the recording in prefrontal cortex with about 3 millimeters away, you can still see a shift from delta to high gamma. It's not quite as profound as locally in this problem, but it's going quite some distance. And in general, the inputs from 6B are very long ranging throughout the cortex.
See, that actually brings us to another question Divya Chander has. Do you think that the layer 6B neurons may give rise to the spindle oscillation we measure during both sleep and anesthesia in EEG? I have many of these recordings and have observed interesting correlations during rapid versus delayed emergence back to consciousness.
Interesting question. So I think I think spindles are thought to be generated by thalamic reticular neurons because of, well, for many reasons. Partly the frequency and some sort of, let's say, entraining that the inhibition of higher order thalamus provides. And this is one place that 6B doesn't project to. So I'm going to say, per se, probably not, but if we're talking about the, let's say, slow or fast emergence of this during a recovery, then I think it could be playing some sort of role. So I mean, one could imagine that the reticular neurons are somehow entraining or pushing the thalamus into some particular rhythm, and that the 6B is let's say urging that in some way, or if it's being pressed, it's lack that. So there could be some interaction on that level, but it's not in the loop per se in terms of the influence of 6B. Nevertheless, it might still, it would be interesting anyway to see how they interact.
We have one more for Matthew from Alessia Bilash, who's actually responsible for patching the dendrite of that neuron in the background. She asks, have you done any experiments exploring the integration of the layer 6B and POM inputs in the dendrites of layer 5 pyramidal neurons, in vitro or in vivo? Do you think specific integration patterns of these inputs would further boost somatodendritic coupling, arousal, etc., more than what you'd expect from optostimulation that you did in the in vivo experiment?
Well, nice question. So on I don't know, and this is a bit awkward because Tim is such a powerhouse that I'm not sure that he hasn't done this experiment. He just hasn't told me about it. Because he has just a whole wealth of experiments that are yet to be, let's say, put together and synthesized. We're in the throes of this at the moment. But anyway, as far as I know, we haven't done that, and I would expect, of course, that it would. One of the kinds of experiments that he has done is to look at whether or not the same of 6B that are targeting the thalamus are also targeting the same places, the same neurons and same tufts and so on. So they seem to be quite conjoined, so that they're forming ensembles, if you like, and so it's not. I don't know. It's like throwing a frisbee now, but yeah, it's not unlikely, I think, that they're going to converge on similar dendrites, and if they would, then I would expect that they'll be synergistic in the way they behave, and that, of course, would be very exciting. What's the second part of this? So, do you think specific?
I think it was.
Yeah.
Sorry, would integration drive better somatodendritic coupling?
Yeah. Yes, and the other thing is that I didn't get a chance to dwell on it for lack of time, but there's it's also inserting into the middle compartment, into layer 5A. The other thing I didn't get a chance to say was that the inputs of the 6B onto the distal dendrites are facilitating, and not the inputs from the thalamus. So there's a different kind of balance going on in the tuft, but it's not that way around for the layer 5A, where the thalamus has quite a large influence, and that's the coupling compartment. So it's going to be very interesting to try to tease out all of these questions, and we're currently in the throes of doing this both in vitro and in vivo, just to further work out, let's say, the dynamics of this circuit. A really good question. I don't know the exact answer, but it's definitely one of the right questions.
Rafa has a question for all of us on the panel.
Yeah, I wanted to be maybe a little bit provocative, and I would argue that there's an elephant in the room, and those are the inhibitory interneurons, no? And that for every one of the three phenomenologies that you've described so beautifully, by the way, incidentally, kudos for the work, the dendritic branchlet activation, the tufts, or the clustering, you could argue that the interneurons are on the driver's seat of that phenomenology, you know? And we heard this morning that more than half of the cell types in the cortex are inhibitory interneurons, and that in fact it looks from the recent data that they have very selective and specific connectivity, and most of them actually target the dendrites. So the suspicion is that they're not just sitting there doing nothing, but they're playing the dendrites like a piano, no? And then the other suggestion that interneurons are really critical, it's actually from our own work. When we look at these dendrites in vitro and in vivo, there's a big difference. And they seem to be operating in vivo under the regime in which most of the time, at least from the voltage dynamics of the dendrites and the spines, they're sort of clamped down so that nothing makes it to the soma unless probably an interneuron wants it to happen. So this is a hypothesis. We haven't demonstrated this. But anyway, this is just a question for you to speculate how the interneurons would interface with the beautiful work that you've presented. Thank you.
You should answer that. Well, I can go first if there's somebody else going. That
I can also take it, Matthew, if you want.
Yep. Okay.
Sure.
Well,
yeah, I mean, it's a great question by Rafa. Obviously, you know, inhibition is an incredibly powerful modulator of dendritic excitability. Idan Segev had a modeling study which showed that even a single inhibitory input can be enough to, you know, at least locally break, you know, the spread of dendritic spikes and the spread of backpropagating spikes. And such inputs can also have, you know, more long-range effects on dendritic excitability. In our study, the dominant effect of stimulating the feedback inputs from LM is actually inhibitory, and presumably that's via some kind of a feed-forward pathway activating local interneurons in the tuft, and that's something that we have to follow up on. We have to identify which interneuron classes are activated by the feedback inputs. That's a great question.
Yeah, and I could add to that also that pretty well every attempt we've made to examine dendritic activity in vivo has initially been flummoxed by one interneuron or other that has basically prevented us examining the activity, and so we have a row of publications on these different interneurons that are having remarkable effects, really spooky, remarkable effects, and like the one Mike was just alluding to. In fact, I think we inherited the postdoc who was responsible for that. Albert Giddon's now in my lab, and he's
Giddon Segev. That's
yeah,
exactly.
And he's working on exactly this question, and it really is, as I say, sometimes very spooky. The other thing to add to that is that amongst the different kinds of inhibitory neurons, there are also a class in layer 1 that are giving GABA B inhibition, and this GABA B inhibition is probably not just inhibiting in the traditional sense, either with hyperpolarization or with shunting inhibition, but also inactivating various conductances like the calcium conductance and the NMDA conductances. And now that adds a whole lot of new complexity to the question you're asking, which I think is right on, that they'd be playing the pyramidal neuron like a violin, I think, in this sense.
Maybe can I add something to that? There are a lot of modeling studies, of course, that have explored the effect of different types of interneurons, and maybe Idan would also like to share with us some of his own. What I found really fascinating when we were building different types of interneurons into our models that we've done with Jahida or with other collaborators was how redundant, let's say, the system is. So if you try to modify one type of an interneuron to take it out, let's say, of the system, others kick in immediately, taking over the same role. And it was very hard in a model, right, to distinguish the different contributions because they would complement each other in such a fast and efficient manner. So I think this is something that is maybe more difficult to dissect experimentally than computationally, but even computationally, it's amazing how complex, an orchestra like playing not just one violin, but all of them at the same time. So it will be a difficult task.
Maybe I can add a final follow-up comment. Speaking as an experimentalist, the biggest missing tool in our toolkit, in our arsenal, is actually a sensor for inhibition. Chloride sensors are not great, and anyway, the chloride signal is very small when you activate a GABA-A conductance, because the chloride gradient is very small compared to, for example, the calcium gradient across the membrane. And the GABA sensors, the GABA sniffers we have so far are not nearly as good as the glutamate sniffers, which have come a long way. So, our molecular biologist friends would really do us a great favor if they could develop a good sensor for inhibition.
It's actually very true. I mean, it's quite incredible how one could think about inhibition sculpting very substructural level computations, right? Like we have the studies from Nelson Spruson showing that it could be even within a branch, different segments are controlled by different GABAergic neurons. And that could probably explain some of the things you saw, Michael, because you are imaging the same dendritic branch. And depending on the proximal or distal end, you could see a very local calcium event, and it's getting shunted so precisely. And then I think the other thing that I often wonder about is how, if there are different pieces of information being carried by different dendritic branches, how is coupling dynamics modulated, that the soma is being dictated by different branches depending on the context. And this kind of goes into also some of the things Jota showed. I actually have a specific question for Jota. What do you think could be, there's this whole idea of representational drift, and you're actually talking about sparsifying representations with the synaptic turnover. And do you think the idea of the representational drift is an actual phenomena or an epiphenomena? And could this turnover play a role in it?
Yeah, I mean, I think it is a phenomenon, but it might be brain area specific. So there are some really nice recent studies showing how we can integrate new information into existing percepts by the addition, let's say, strengthening of new spines. And this at the end looks like representation drift because it's not exactly the same as the original memory, but it is in fact an update of the original memory. So that could be an explanation as why there is a drift, but it's not that the first memory is eliminated, but it's now becoming a part of an updated representation. And this is probably more pronounced in areas like the hippocampus, where memories are supposed to live for a shorter time period versus, cortical areas. And I don't know if people have compared this across different brain areas, but I think synaptic turnover is definitely, a strong advantageous mechanism for incorporating new memories. But if you have too much of it, of course, it could also lead to rapid forgetting. And there, I think there are also studies showing that animals with increased synaptic turnover can also forget faster. So, there are two sides of one coin. It could be a positive or a negative effect. By the way, can I ask one question since I am talking with respect to interneurons? I wanted to ask Rafa. I've always been wondering, we have such a wide variability in the molecular profiles of the different types of interneurons. Do you experimentalists people think that they have also different functional roles, or is there some kind of, let's say, redundancy in the type of interneuron as described by its molecular profile, which is not evident at the functional level? Or otherwise, why do we need so many different kinds of interneurons? What's the point?
Yeah, it's a great question. And I mean, this is another elephant in the room. Why do we have so many cell types, not just interneurons? I mean, the pyramids are also, why do you need 60 cell types of pyramids if they're only doing one job or maybe 10 jobs? I think the answer is, of course, we don't know, but the types of tools and atlases that are being developed by the Allen Institute and other groups hopefully will let us tackle that question. But the normal answer, going back to Cajal, and particularly anchoring the beautiful work that was done in the retina, is that different cell types not only have different morphologies and different connectivities, but different circuit functions. So in the retina, 30 different types of retinal ganglion cells, and almost every one of them has a different function. So you could just extrapolate, like, well, maybe the cortex is different, but maybe it is like a retina, in which case the prediction would be that they're not just redundant, that they all have a job. And if you look at some of them, there's some super specific patterns of axonal projection and connectivity, which speaks to the fact that they have to have a very specific job. It would not make sense for nature to build something which has specificity at the structural level and not use that at the functional level. It would be a complete dissociation. Why would you want to do that? So yeah, this is again another elephant in the room. And I mean, some of the work that we heard this morning and this afternoon, it's starting to dig in that direction.
Mm-hmm.
Can I add one word about inhibition?
Yeah.
Go for it.
So I think I was also puzzled, still am, about this very large number of inhibitory cell types, but I know that they are targeting many times different parts of the dendritic tree. So the basket cells tend to more target nearby, near somatic region, and let's say Martinotti tends to go more far away in the more distal parts. And so I look at them today as a breakers, as a stoppers of local nonlinearities. So if you want to, let's say, stop local nonlinearity in the apical tuft, you will probably activate a Martinotti cell. If you want to activate to stop more nonlinearity event in the basal dendrites, you will use probably maybe the basket, and then you can use the chandelier to block everything. So it's a hierarchy of inhibition. And you can use this hierarchy, as Mike knows very, very well, because in the deep neural networks today, there is a whole tendency to add dendrites and inhibition. As you know, most of the deep neural networks do not use inhibition in a specific way, rather than a feed-forward, so to speak, in excitation. But the modern style of deep neural networks are now adding dendrites with inhibition, because you can gate, as Mike showed with Peter Latham and others, you can gate information on one, so to speak, channel of dendrites, while you allow information to flow in the other dendrites of the same neuron. This gives you a lot of much more capacity, memory capacity, but also, the preferential control on what you want to learn or not. So I think that the deep neural networks are starting to add this complexity with inhibition, because you can gate information in an interesting and in non-linear ways.
Mm-hmm.
But you don't need 32 types Idan to do that. 3 types will do, or 4 types will do. That's what puzzles me. Why so many?
I think one of the things we could think about is like something like Matthew alluded to. So there's this behavioral state dependent modulation, right? And then these fine scale subcompartment level coupling dynamics could be modulated in a behavioral state dependent manner. And then all these different neuromodulatory systems kick in. And, it's not just a binary 0 or 1 on off switch. It's sort of a whole, depending on the sensitivity of the interneuron type, like VIPs are very highly sensitive to like cholinergic modulation. Somatostatins are also, but at a slightly different level or lower level. So it gives us this whole dynamic range probably depending on like the neuromodulatory state. Obviously, maybe 32 is overkill, but I kind of feel like given all the types of information and different contexts that the system has to encode, you'd probably want to design a system where you have a very nice graded scale and titrate it to different maybe levels of arousal or levels of salience or a whole host of features that you need to encode.
That's a great point. And in addition to differential neuromodulation of different interneuron subclasses, it's important to remember that many of these interneurons actually release other neurotransmitters in addition to GABA, like VIP, substance P, somatostatin, and there may be functions of these neurotransmitters that are, still completely unexplored, right? Maybe a whole additional layer of, importance of these different classes that depend on these co-released neurotransmitters.
Yep.
I also want to add that with the inhibition, it's not just a matter of where they're targeting the neuron, but I think that something that, I think requires some thought also, from modelers is the fact that different interneurons, because of their firing patterns, may have really different types of effects. It's not like all inhibitory synapses are necessarily equal in terms of their shunting capacity. And even though at the moment people seem to think that most of the dendrites are targeted by some interneurons, we're actually seeing that that's not necessarily the case, that there's actually a lot of PV innervation also on the dendrites. And that has a really very different sort of postsynaptic effect. And I kind of wonder whether you can model something like that in terms of these different types of synapses and their kind of ability to, in terms of range and spatial and temporal range of their sort of shunting effect. Yeah.
Absolutely. Ellie, that's, in a study we've done a few years back, where in the prefrontal cortex, where we used fast spiking interneurons targeting the cell body, and then, like some light interneurons targeting the dendrites. And then we switched their targeting location just to see the effect at the cell body. And clearly it is very different, right? So it's not only where it projects, but it's how it spikes. The firing pattern makes a big difference on the effect at both the dendrite and the cell body. And, it's interesting and definitely feasible with modeling to explore the relative contributions. So, yeah, Dan has done something like that as well, I think, in the past.
I think Rafa had a question for, sorry, go on.
I was just going to say the corollary of what Yoto was just saying, and what everybody's been saying. It's kind of redundant in a way here, but I think it's important that I don't think we have a hope in hell of explaining what all this diversity of interneurons does in terms of the circuits without including the dendritic properties, with the kind of point-to-point diagrams that one normally sees of the cortex. There's no way, I think, to include the diverse effect of interneurons.
Right. Rafa, do you want to ask your question?
Well, actually, Jayita, since we're pretty much out of time, if it's okay with you, I would just forget about the question and maybe make some final comments in deference to the time and attention of our attendees that have been patiently watching this for most of the day today.
Yeah.
So,
there are a couple more questions in the Q&A, which feel free to just type in the answer for them then, since we're running out of time. Rafa, did you want to do your concluding remarks now?
Sure. Yeah, anyway, so I thought that just personally, this was a great meeting, very stimulating, and it speaks to the issue that this is foundational work, whether you look at this circuit from the dendrite point of view or you look at the dendrites from the circuit point of view. I mean, it becomes clear from these types of discussions and data we've heard today that you cannot understand one without understanding the other. No? And then, so this would be maybe one take-home message that we're not going to figure out how the brain works. We cannot take apart these neural circuits one cell type at a time without understanding what the dendrites are doing, and vice versa. We cannot just look blindly at the dendrite without thinking about their circuit function. Another take-home message is that there's a lot of open questions. We heard a lot of examples today. I was personally very excited by every single talk, with beautiful science of, and this speaks to all the other open questions that are there. No? So this is an area that has a lot of gold to be mined, which means that's a lot of work to be done. And putting a little brick on that work that needs to be done, I'm excited to work in this team Dendrites with these 5 other labs that you've heard about, thanks to the NINDS. But I wanted to use this opportunity to bring everyone in the audience in, and of course all the speakers. This is a job for that we're going to need everyone's help to figure out how dendrites work and how circuits work, you know. Our team will try to help by providing tools that we're going to be building, from molecular biology tools, just answering Michael's prayer for a sensor for inhibition. This is exactly what Michael Lin is going to try to deliver, or at least to measure optically IPSPs at the dendrite. No? Maybe also we'll be able to deliver, besides molecular constructs, mice, and of course software, and even possibly hardware to how to play the violin with the dendrites with using holography. We will hopefully also provide and shared data from all of our different 6 labs approaching the dendrites, again, of one single cortical cell type, because in this jungle of cell types, the last thing we want is to get lost in the jungle. We'll just stick into one and see if we can understand one cortical cell type. No? And we will provide this data in a website that we're soon going to be making public, that will serve as our interface between our team of 6 labs and the rest of the world, the rest of neuroscience. And again, also with an annual meeting where we can focus on that could serve as a watering hole for the community of people, that the nerds like us that care about what the hell do dendrites do in the middle of a cortical circuit, in the middle of a circuit. And also, hopefully, with engaging in direct collaborations with some of your labs, not just from the speakers, but also from the attendees, the audience. Know that. So our door is open, our hand is open, and hopefully through you'll find us through the website, and if not, you can just email us directly. No? And just wanted to finish by thanking the Chen Foundation that generously has helped us with this meeting as part of a series of meetings at the Neurotechnology Center, who also I wanted to thank for support, and of course our patrons for this RM1 grant, the combined program of the NINDS, for their generosity and their trust in this crazy group of people who want to shake out the dendritic tree. And finally, I wanted to thank Lisa Gibson, who hopefully can turn on her camera, who's the person, the organizer of the meeting, who's been flawless in their execution of the IT and the entire meeting. Lisa, has been such a pleasure to work with you, and you deserve all the credit for this. And that pretty much sums it all. I think I wanted to wish everyone a good evening or a good night. Thank you, the Europeans, for staying up so late, and the Israelis even more. And we'll see you soon. Thank you.
Thank you. Thank you.